Good morning, Good morning, everyone. This is Malar Kamin on behalf of PAA, and I welcome all the students and faculty for this webinar. We are glad to do this webinar for the students. Today, I'm very happy to introduce Dr. A. Sumathy. She's the head of, she's currently in Periyar College of Pharmaceutical Sciences. She's completed a bachelor's, master's, and PhD from Periyar College. She's a hardcore Periyar College alumni, I would say. And she's been with Al Shifa College of Pharmacy, Kerala previously, and she's currently the professor and head of the Department of Pharmacy, Srinivasan College of Pharmaceutical Sciences in Trichy. To her credit, she has 2 patents, and she's been awarded the Best Innovative Ideas of Research Award. She's won the first prize as a cash award, and she has a lot of laurels to her. And we are very happy. She's also published a book entitled Pharmaceutical Product Management. So we are very happy to have you, ma'am. And I think you can go ahead and deliver your lecture.
Thank you very much for introducing me, sir. And also on behalf of myself and my management, I would like to thank for providing me an opportunity to interact with the students. So thank you very much to one and all. And now I am directly getting into my session on microbiology. So shall I share my screen, sir?
Yes. Please go ahead, ma'am.
Yes, okay, thank you. So today, being Sunday, many were busy with many things. But again, I would like to congratulate PAA-MMC and IPGA for providing a great platform to the students to utilize their Sunday for making their GPAT coaching class. So thank you very much once again. And today we are getting into the microbiology topic. Okay. So microbiology, again, the GPAT syllabus covers the following titles, what I have listed here: Introduction to Microbiology, Microscopy and Staining Techniques, Biology of Microorganisms, Fungi and Viruses, Aseptic Techniques, Sterilization, Disinfection, Microbial Spoilage, Vaccines and Sera, Microbial Assay. Okay, so today I'm going to briefly discuss about biology of microorganism. Especially, I'm going to dwell with the bacteria. Okay, so before getting into the bacteria, I would like to give a few introduction about microbiology. Then we can directly move on to bacteria classification and other things related with bacteria. Okay. So coming into the microbiology, as you all know, microbiology is nothing but study of science that related with the microbes. Again, microbes are the things that we could not able to see, are able to visualize with the help of naked eyes. It needs some special equipments like microscopes to visualize and to study the things related with the organisms. So those organisms are called as microbes, and the study of those microorganisms are called as microbiology. Okay, and basically this microbiology or this microorganism are the whole living organisms are classified into various categories. So under GPAT syllabus, you can find the first title as Whitaker 5 Kingdom Classification System. Okay, so Whitaker 5 Kingdom Classification System, before getting into that, basically it is derived into 2 basic types. Okay, there are 2 major kingdoms. Initially, the 2 method of classification was given by Linnaeus in the year 1735. In the year 1735, Linnaeus classified all living organisms into 2 major kingdoms. Okay, so the 2 major kingdoms are nothing but plants and animals, and it is called by the biological term Plantae and Animalia. So these plants and animals are categorized based on very important criteria: presence of RBC. Okay, so based on the presence of RBC, that is the red blood corpuscles, 2 major groups are categorized, which is nothing but plants and animals. Okay, and the major difference between this plant cell and animal cell, you might be studied in your higher classes itself: presence of chloroplast and presence of cell wall, and these 2 things are not present in animal cell. Okay, so this is the first system of classification, 2 kingdom classification system. Okay, from 2 kingdom classification system, then the system is moved on to 3 kingdom classification system. So 3 kingdom classification system is nothing but, apart from considering their biology, here Ernest Haeckel, he is the scientist who considered the mode of nutrition. So how they prepare their food, how they are living. So based on their mode of nutrition, or we can tell mode of feed, they are categorized into 3 kingdom classification system. So from 2 kingdom, now it was shifted to 3 kingdom classification system. So here they consider the mode of nutrition. So mode of nutrition, either they can prepare their own food or they can depend upon the other living organisms for their food. So apart from these two, the third category comes the saprophytes, which utilize the dead and decaying materials for their feed. And this category is broadly derived as protista. So now comes 3 kingdom classification system, plants, animals, and another group called as protista. And this protista includes algae, fungi, and protozoa. Okay. So again, apart from these 3 kingdoms, the 2 major cell groups are present. Okay. One we can call it as eukaryotes, and another one we can call by the term prokaryotes. Eukaryotes and prokaryotes. So 2 major type of cell groups, and this is based on the level of cellular organization. Okay, so how the cellular makeup is present, how the cells are managed or arranged or optimized together. So based on this, they are categorized into 2 major category. One is eukaryotes and another one is prokaryotes. Okay, so eukaryotes, the term eu, that is EU, which is nothing but true or pure. So here the eukaryotic organisms, they have a well-defined membrane bound organelles. So what are all the cellular components present inside the cell? They consist of a well-defined organisms. Okay, they consist of a well-defined membrane. So they are called as a membrane bound organelles, and those category are coming under eukaryotes. Okay, say for example, human beings, plants, fungi, and insects. So these are categorized under eukaryotes. And coming to the another group, prokaryotic cells. So prokaryotic cells are nothing but they do not contain a nucleus. We can call them, they consist of nucleoid material. They lack membrane bound organelles. Okay, they do not have a distinct or definite membrane to segregate each components which is present inside the cell. So very best example is bacteria. And here you can see a very simple representation to view prokaryotic cell and eukaryotic cell. So here the nuclear membrane is present and no such membrane is present in case of prokaryotes. Okay, and here comes the difference between the basic difference between the prokaryotes and eukaryotic cell. So in case of prokaryote, it consists of a circular DNA and no organelles are present, no membranes are present. It consists of single chromosome. And another,
ma'am, sorry to interrupt you.
Yes, sir.
Your slides are not changing, ma'am. Check pandringla konjam.
But for me, it is changing. Where it was, sir?
It's in the first slide, microbiology, introduction to microbiology. That's the only one that's stuck. Maybe you close panni thirumba open panni parunga.
Okay, sir. Sure, I will.
Sorry to interrupt.
No problem, sir. Okay, sorry.
And the students text panniranga, so I just
okay, sir. Okay. Sir, is it visible now, sir?
Yes, ma'am. Or two slide change panni paathu, then you can go back to your yeah, it's changing now, ma'am.
Okay, sir. Sorry.
Okay, you can go back to your the one which you are
okay, sir, sure. Now I'm started discussing about difference between prokaryotes and eukaryotes.
Okay, ma'am. Yeah.
Okay, so prokaryotes, again, it consists of a circular DNA and no organelles are present and no membrane bound nucleus is present. It consists of single chromosome. Okay, and another major criteria to differentiate prokaryotes and eukaryotes, here it consists of, in case of eukaryotes, it consists of many number of receptors present on the surface of the plasma membrane. Okay, so in the surface of the plasma membrane, it consists of receptors through the receptors only, whatever things which want to enter into the cell, it will go and interact with the receptor. After binding with the receptors, then it is allowed or get inside the cell. Okay, so these things are not present. So this surface receptors are not present in case of prokaryotes. Okay, and in case of prokaryotes, it consists of a complex cell wall. Majorly, it contains peptidoglycan molecules. Whereas in case of eukaryotes, it consists of a very simpler components. In case of fungi, it consists chitin, and in case of plants, it contains cellulose. Okay, and if you come into the transcription and translation process, both occurs simultaneously. Okay, so transcription, again, nothing but the process of converting the DNA into mRNA. Okay, so here the mRNA coded proteins are synthesized by translation process. So both occur simultaneously in cytosol in case of prokaryotes, whereas it occurs separately in nucleus, and translation occurs separately in cytosol in case of eukaryotes. Okay, and the composition of flagellum is different, and it consists of haploid genome. In case of eukaryotes, it consists of diploid genome. Here it have plasmids, and in case of eukaryotes, plasmids are absent. It is not very common. Very rarely we can notice plasmids. And again, what do you mean by the term plasmids? Plasmids are nothing but it is an extra chromosomal genetic material. Usually the genetic material or genetic information is embedded only in the form of chromosome that is inside the nucleus. Okay, so inside the nucleus we supposed to find the chromosomal or genetic material that is responsible for passing the genetic information from one generation to next generation. Okay, whereas these plasmids are nothing but they are called by the term extra chromosomal genetic material. Okay, so this extra chromosomal genetic material they utilized to transfer. Okay, so during we are having the another part, genetic transformation. So that is transduction, conjugation. So in those process, the bacteria or other microbes, they tend to share this extra chromosomal genetic material. Okay, so instead of sharing their chromosomal part, that is their own genetic material, apart from the genetic material present in nucleus, they supposed to transfer this extra chromosomal genetic material from one bacterium to another bacterium or one microbe to another microbe. Okay, then it consists of small ribosomes, no histones are present, and also it lacks cytoskeleton. Okay, the cell ranges from 0.5 to 100 micrometer in case of prokaryotes, whereas eukaryotes 10 to 150 micrometer. And prokaryotes, mainly it will undergo reproduction by asexual, that is binary fission. In case of prokaryotes, it undergo sexual reproduction, that is meiosis and mitosis. Okay. So next comes 5-kingdom classification system. So 5-kingdom classification system, it includes Monera, Protista, fungi, plants and animals, that is Plantae and Animalia. And here out of this 5-kingdom classification system, so now we have added up Monera and fungi. Okay, so initially under Protista, this fungi is also placed. Okay, so now they got a separate kingdom by the intervention of Whittaker in the year 1969. Okay, so here comes the full picture. So based on three criteria, this 5-kingdom classification was happened. So what are all the three criteria? So first one, based on the cell type, either it is a prokaryotic cell or it is a eukaryotic cell. Okay, so based on the cell type, either it is a prokaryotic cell or eukaryotic cell. And second comes the level of organization. And again, level of organization, nothing but either it is occurring in colonical form or solitary form, that is individual or in the form of colonies. And another one, either it is in the mode of unicellular or multicellular. Okay, and the third criteria comes based on their mode of nutrition. So how they are producing their feed. Okay, so three criteria based on the cell type, based on the level of organization, and based on the mode of nutrition. So based on these three criteria, the whole living organisms are again pooled under 5 different kingdoms. Okay, 5 different kingdoms. So to have the list, Monera, Protista, Animalia, Plantae, and the last one is fungi. Okay. And before this Whittaker classification system, and again we have another one, 4 kingdom classification system, under which fungi is not having a separate place. Okay. So under 4 kingdom, Monera, Protista, Animalia, and Plantae. Okay. So this is coming under 4 kingdom. Okay. So again, I repeat. So initially 2 kingdom classification system, only plants and animals based on their either RBZ is present or RBZ is absent. Okay. And second comes 3 kingdom classification system. And this 3 kingdom classification system, apart from plants and animals, they added 3rd kingdom, which is nothing but Protista. And this Protista arises because of the mode of nutrition. Okay. And comes the 4 kingdom classification system, which includes Monera, Protista, Animalia, and Plantae. Okay. And finally comes the 5 kingdom classification system, which includes fungi also. Okay. So here in this chart, you can get a better idea. So the organisms are basically classified into prokaryotes and eukaryotes. Okay. So, and again, under prokaryotes, we can have only one group. All prokaryotes are pooled together under one criteria or one kingdom, which is nothing but Monera. Okay. And eukaryotes, and again eukaryotes, we can have unicellular and multicellular. So if it is a eukaryotic unicellular, it is placed under Protista. It is placed under Protista. Okay. And comes multicellular. Under multicellular, again, we have 2 different sets, one which contains cell wall and the other which is not having the cell wall. Okay. So if cell wall is present, then it is categorized under the group, under the kingdom Animalia. Okay. Then one which is having cell wall, okay, which is categorized under based on the mode of nutrition. So with cell wall and multicellular eukaryotes, if they prepare their own food, then they are called as phototropic. Okay. So phototropic are nothing but they can able to prepare their own food. And heterotropic, and again they depend on other feed materials for their food. So phototropic, which is nothing but Plantae kingdom, and heterotropic, which is nothing but fungi kingdom. Okay. So now comes 5 major kingdoms: Monera, Protista, Plantae, fungi, and Animalia. Okay. And here another summary chart showing what are all the different characteristic feature of these 5 major kingdoms. Okay. So Monera, unicellular prokaryotes, and Protista, unicellular eukaryotes, fungi, multicellular eukaryotes, and plants, multicellular eukaryotes, and animals, multicellular eukaryotes. And here this is with cell wall and without cell wall. Yes, sir, anything else to be you want to say from your side?
Nothing, ma'am. All okay.
Okay, sir. Okay, thank you. So here you can also able to see what are all the different examples for this category. Okay. So for Monera, we can find bacteria, blue-green algae, and for Protista, amoeba, paramecium, and eugenia. And for fungi, yeast, rhizopus, mushrooms. And for Plantae, trees, plants, and shrubs. Okay. The same way for Animalia, fishes, insects, animals, and of course, we the human beings, birds. Okay. And here another mode of representation. So the same thing I am presenting in different views to make you to understand in a better way. Okay. So here starting from the base, Monera, we are having this prokaryotic cell. Then comes the another cell line, that is protist. And again, 3 major groups: plants, animals, and fungi. Okay. And here the overall summary chart of 2 kingdom classification system, which was given by Carolus Linnaeus in the year 1758. And 3 kingdom classification system, it is given by Ernest Haeckel in the year 1866. And 4 kingdom classification system, it was given by Copeland in the year of 1966. And finally, 5 kingdom classification system, which was given by Whittaker RH in the year of 1969. Okay. So what are all the kingdoms under each title that already we summarized? Okay. So now moving on to another hypothesis. Okay, so for this hypothesis, we are going to describe about 3 domains. Okay, so before getting into the kingdom, that is hierarchy to the kingdom, it consists of 3 major domains. So how this 3 major domain comes? So here we should know about the term called LUCA. So what do you mean by the term LUCA? LUCA is abbreviated as last universal common ancestor. So ancestors, generally we can tell our pre-fathers. Okay, so those that is primitively derived cells. So it is described as ancestors. So here various theories, various hypotheses are already existing based on this prokaryotic and eukaryotic classification. Okay, so so many theories are in existence, but the common theorem or common hypothesis is all cells came from a common ancestor. Okay, all cells are derived from the common ancestor, which is nothing but that is the one which present while the world or universe has been started. Okay, so that common ancestor is called by the term LUCA. Okay, so this LUCA is divided or evolved into different domains, and these 3 domains are represented as bacteria, archaea, and eukarya. Okay, and again this bacteria and archaea is nothing but they are belonging to the group prokaryotes. Okay, so we divided into prokaryotes and eukaryotes. So now again this prokaryotes are clearly putting under 2 different domains, which is nothing but eubacteria and archaea. And this eubacteria and archaea, they are differing in few characteristic feature that I will discuss with you. Okay, so eubacteria, and again this eubacteria is nothing but they are defined by the term true bacteria or peptidoglycans, because their cell wall majorly consists of peptidoglycan. Okay, and archaea is nothing but odd bacteria. Okay, even though it consists of major characteristic feature of bacterial cells, but it is different from the bacteria. Okay, that's why it came under another domain called as archaea. Okay, and the last one is eukarya. Eukarya is coming under the eukaryotic cell types. Okay, so here you can able to see the phylogenetic tree. So under the phylogenetic tree, you can able to see what are all the things categorized here. We can call them as phylum or subphylum. Okay, so these 3 are the major 3 domains. So under these 3 domains, we have 5 kingdoms, and these are all the various phylum present under the 3 domains. Okay, and now coming to the difference. Okay, if you look into this picture, you can able to clearly remember about the difference. Okay, so on the left hand side, you can able to see the membrane lipids present in the bacteria and eukarya, whereas on the right hand side, you can able to see the membrane composition of archaea. Okay, so here the cell wall composition, mainly it involves the hydrocarbon chains. Okay, so you can able to see this mountain like structure or triangle like structure. It represents the hydrocarbon chains. Okay, so this hydrocarbon chains are branched. Okay, here you can able to see one more line, one more dropping line in case of archaea. So in case of archaea, it consists of branched hydrocarbon chains, whereas in case of bacteria and eukarya, these are unbranched hydrocarbons. Okay, and once again, these hydrocarbons, they are attached with the glycerol molecule. Okay, so while attaching with this glycerol molecule, here you can able to find ester type of linkages present in case of bacteria and eukarya. Okay, whereas here it involves the ether linkages. Okay, so ester linkage is present in case of bacteria and eukarya, and ether linkage is present in case of archaea. Okay, so this is the major difference which present in their structure and composition. Okay, and another thing, in case of archaea, there is no peptidoglycan. Peptidoglycan is absent in case of archaea, and these archaea are not sensitive to antibiotics. Okay, these archaea are not sensitive to antibiotics, whereas in case of bacteria, bacteria is immediately treated with the help of antibiotics. Okay, so archaea are resistant to antibiotics, whereas bacteria, they are sensitive to antibiotics. Okay, and now comes why they can often withstand this extreme environment. Okay, compared to bacteria, the archaea can able to withstand extreme environmental condition, and this extreme environmental condition is because of the presence of their presence of their linkage. So what type of linkage is present in archaea? Archaea consists of ether linkage. So this ether linkage helps the archaea to withstand better in case of extreme environmental condition. Okay, whereas the ester linkages, compared to ether linkage, the ester linkages break down much faster. Okay, so that's why this archaea can able to live in extreme environmental conditions. Okay, so bacteria, which is also called by the term eubacteria, are true bacteria. Eubacteria are true bacteria, and again we can have both beneficial and harmful rules. So if you go look into the harmful rules, it is responsible for causing a variety of diseases. Okay, still we are fighting with a variety of diseases, and also we are counteracting with those diseases by our treatment schedules. On the another side, the beneficial role of bacteria, it is very much helpful in various type of rDNA technology, in the production of enzymes, in the production of vitamins, and many solvents, in the production of many industrial solvents. Many more beneficial roles also happens with the help of bacteria. Okay, and eukarya, the eukarya and bacteria, they can also have many similarities, and also they can have many differences. Okay, so what is the similarity between bacteria and eukarya? So both have unbranched hydrochains, and both consist of what type of linkage is present here. So here it consists of ester linkages. Okay, so this is about bacteria, eukarya, and archaea. So here comes the summary chart to represent what are the cell types present here, what type of chromosomes are present here, and different types of ribosomes, and ribosomal sequence. Okay, what is the sequence which is present in ribosomal RNA? Okay, so in all the three cases, it consists of positive ribosomal sequence, but even though it consists of positive sense rRNA, but the rRNA is unique with bacteria, archaea, and eukarya. And what do you mean by the term unique? It consists of a specific molecular pattern. Okay, so the molecular pattern which is present in archaea is unique only to archaea. We could not able to predict the same in case of bacteria and eukarya. Okay, it won't repeat in other two cases. The same way, rRNA is unique and specific to bacteria, and rRNA is unique and specific to eukarya. Okay, you can call it as a specific molecular region or specific molecular patterns. Okay, and peptidoglycan is present in bacteria, whereas it is absent in archaea and eukarya. And sterol membrane, and again sterol membrane is present only in eukarya, and it is not present in all type of bacteria, but few bacteria contains this sterol membrane. So few exceptional cases are also present. And this is a picture you can able to find in your reference book, that is Prescott. Okay, microbiology, Prescott's microbiology, and here it gives a full view. So in the left hand side, you can able to see prokaryotes and eukaryotes. Okay, how these two cells are categorized, and under prokaryotes, what is the three domains? Two domains we have discussed, archaea and bacteria. Okay, and the third domain, eukarya, it is coming under eukaryotes. Okay, so I hope it is clear. You can able to see three domains together. Okay, so archaea, bacteria, eukarya. So three domains are represented together. Under these three domains, archaea and bacteria is coming under prokaryotes, whereas eukarya is coming under eukaryotes. Okay, and also you can visualize the five major kingdom, which is highlighted with blue color. So this five major kingdom. So first kingdom is Moneria. So this Moneria, again, it coming under prokaryotes, and another one is major four kingdoms. Other rest four kingdoms is falling under the category of eukaryotes. Okay, and you can able to see two cell type, which is nothing but prokaryotes and eukaryotes. Okay, so this is the summary or gist of the classification of microorganism. Okay, and here you can able to see, and this is the one you can also get it from Prescott's microbiology. Here the comparative characteristic feature of bacteria, archaea, and eukaryotes, you can get it in a clear manner. Okay, and now to end up with classification, the very important thing is taxonomy. So what do you mean by taxonomy? Which is nothing but organizing classifying and allotting name to all living beings. Okay, so first it is organized, it is put under a particular group, then it is segregated or classified, then finally it is allotted with a name. Okay, so this formal system of classification was given by Carolus Linnaeus. Okay, so this classification system is called as binomial nomenclature. Binomial nomenclature, that is modern system of naming the organism. So by means of introducing this binomial nomenclature, Carolus Linnaeus is known as the father of modern taxonomy. Okay, so this it includes what are all the cadres present under taxonomy. So domain. So according to domain, what are all the domains we have seen? Three major domains we have discussed. Okay, so what are the three major domains? Bacteria, eukarya, archaea. And next comes the kingdom. So kingdom, 5 major kingdoms. So Moniria, Protista, Fungi, Plantae, and Animalia. Okay, and under phylum, it is coming under the phylogenetic tree. The list will be going on. And the next comes class, order, family, genus, species. And this last two, genus and species, it is represented as the binomial nomenclature. It gives the botanical, zoological, or biological name of the living organism. Okay, and here is the picture. You can see the Carolus Linnaeus. Okay, and this is the naming and how a name of the microorganism to be written. Okay, the first part it indicates the genus name, and second part it indicates the species name. Okay, first part it should start with capital letter, and second part should start with small letter. Okay, and the both part should be written in italicized. Okay, while go for typing, it should be in italic form. Okay, and here comes three major types. Again, under species, we are having a different types. Okay, so species are nothing but the overall, it consists of a similar pattern of triad. Species are nothing but overall it consists of similar pattern of triads. So similar pattern of triads means it consists of genetically determined pattern is similar. It is having a same genetic mode of information. So that we can call it as triads, genetically same determinants. Okay, whereas strain or variety, to understand better, now I can quote the example of coronavirus. Okay, so coronavirus, and again it consists of different strain, or we can call it as different variety. And delta variety, now we can have delta variety and omicron variety. Okay, so these strain or variety is indicated, which is nothing but they are the culture derived from a single parent. They are the culture derived from the single parent, but they differ in their structure and also in metabolism. Okay, they are having different structure and also different metabolic activities. Then they are called as different strains or different variety. And last comes type, or we can call them as subspecies. Okay, so these types are subspecies. The main thing to be remembered is they differ in their pathogenicity. Okay, so because of difference in pathogenicity, which is nothing but the antigenic makeup. Okay, the antigenic setup of the individual of that particular microorganism is different. Okay, if the antigenic setup is different, and again this antigenic setup is mainly responsible for causing the disease, and also it is responsible for deciding the severity of the disease. Okay, whatever the antigenic setup may be, if it is coming under same species, it's supposed to cause a particular disease. Okay, the disease might be same, but again the severity of the disease we can call as a pathogenicity. Okay, so how severe the disease may be, whether the disease stops only by including mild symptoms or severe symptoms, or it may lead to acute symptoms, or sometimes it may lead to chronic symptoms. Okay, so depending upon this severity, and again the severity depends upon the antigenic makeup of that particular microbe. Okay, so they are categorized as the subspecies. Okay, so species, strain or variety, and the last one is subspecies. Okay, so this is about the classification of microbes. Okay, so to summarize under classification, so we discussed about 2 kingdom, 3 kingdom, 4 kingdom, and 5 kingdom, and 3 major domains. What are all the 3 major domains? Okay, and taxonomy. Okay, is it clear? Okay, so now we are moving to the next part. Okay, So the next part under which we are moving to the bacteria. Okay. So now coming to the biology of microbes. Okay. I hope you can able to see the slides bacteria. Yes.
Yes, ma'am.
Okay. Thank you, sir. So under biology of microbes, so I'm going to discuss with you briefly about what are all the different classes of bacteria, and after that, if time permits, just we can have a look on the biochemical test. Okay. So now comes to the bacteria. So bacteria are microscopic prokaryotic unicellular living organism. Okay. So here in the definition itself, you can clearly define under what category it comes. Okay. So it is a type of prokaryote. All the prokaryotes are unicellular in nature, and it is found everywhere. Okay, and it is found everywhere, and it can be harmful or beneficial. So as I already mentioned, and below you can able to see the structure of bacteria. Okay, and while looking into the structure of bacteria, it consists of the cell wall, and the cell wall is followed by the capsule, and inner to the cell wall we have a cytoplasmic or plasma membrane. Okay, and here you can able to notice two main protrusions. Okay, so one protrusion, flagella, which is extended outward from the inner membrane. Okay, so from the cytoplasmic membrane or from the plasma membrane, it protrudes outside, which is nothing but flagella. And this pili, we can call them commonly as sex pili. So this pili are the projections which are projected from the cell wall. Okay, which are projected from the cell wall, or sometimes if capsule is present, the capsule is the outermost layer. Okay. So now classification of bacteria. So the bacteria is classified based on different criteria. So the major criteria, shape and arrangement, and another one is composition of the cell wall. Okay, shape and arrangement, and also composition of the cell wall. So this apart from shape, arrangement, and composition of cell wall, we can have some other criteria to categorize this bacteria, and the same criteria is coming under nutritional requirements also. Okay, so the nutritional requirements, and again to promote the growth of optimal growth of the microbes, what are all the things to be managed? So for that nutritional requirements, this classification factors is also considered. Okay, so here I have pulled together what are all the criteria to classify bacteria, and what are all the criteria to be considered to promote or to reach the optimum growth. Okay, or we can call them as the cultural characteristic. Okay, so first we are moving to the morphological characteristic feature. Okay, so under morphological characteristic feature, so three main morphological characters are present: cocci, bacilli, and spirals. Okay, so what are all the three main or common microscopic morphology? Cocci, bacilli, and spiral. So cocci is nothing but it is spherical or ovoid in shape. Bacilli, rod shape, and spirals. And again under spirals, we can able to see three different forms. Okay, apart from this, we also have three miscellaneous category: rickettsia, actinomycetes, mycoplasma. Okay, so before getting into the basic shape, I want to mention about the miscellaneous category. Okay, so this miscellaneous category, rickettsia is nothing but it is having all the characteristic feature of bacteria, but it is placed under another group called rickettsia. So this rickettsia, though it consists of nuclear material, that is both DNA and RNA, but it is in need of a living cell. It is in need of a host cell to survive. Okay, but we cannot place this under virus group because it contains both genetic material. Okay, so that is why it has got a separate cadre. Okay, and next comes actinomycetes, and another one is mycoplasma, and here they have abnormal morphological features. It is having all other characteristic feature of bacteria. Apart from that, it is having abnormal characteristic features. Okay, so actinomycetes, it is having a ray-like structure. That's why it is referred by the term actinomycetes. And mycoplasma, morphologically it is differing from bacteria, but all other characteristic features are similar with the bacteria. Okay, so now coming into the cocci. So cocci, it is derived from the term coccus. Okay, it is derived from the term coccus, which means berry. So you might be seen about berry. So here I have given you the pictures of berry. So strawberry, blueberry, blackberry, bilberry. Okay, so berry, it is derived from the term coccus. Okay, so based on the number of berries, okay, based on the number of microbes, so again it is classified into monococci. Okay, so mono means single. So if it consists of only one berry, only one cocci, then it is called by the term monococci. So example, Micrococcus flavus. Micrococcus flavus. Here the examples are very important. Okay, then if they occur in pair, that is one berry is occurred along with another berry. Okay, if they are occurring in pair, then it is called as diplococci. Okay, the example is Streptococcus pneumonia. And then comes the third one, chain. Okay, if they are occurring in chain, that is one attached to other with a line, like a train compartments. So one after the other, if they are present, then it is called by the term streptococci. Okay, then another group, if they are occurring in group together. Okay, and while occurring together, again occurring together in different forms. If they are occurring as a group, that is tetras, 4 in a group, then it is called by the term tetracoccus. If it consists of 8 in a group, then it is called by the term sarcina. Sarcina. Okay. Then if it occurs as a cluster, okay, that is a bunch of grapes. If it occurs as a cluster, then it is called by the term staphylococci. Okay. So many numbers are joined together in the form of cluster, staphylococci. Many numbers joined in the form of chain, then it is streptococci. Okay. So this is about cocci. Then here you can see this is micrococci, that is single berry. Diplococci, 2 berries are connected together. That is berry. I am telling about the microbe. Okay, so here streptococci joined in the form of chain. Here tetrad. Tetrad is nothing but occurring in group. 4 occurring in group. Okay, this is sarcina, 8 occurring in group. So in the picture you can able to see only 7 and another one at the back side. Okay. And here staphylococci like a bunch. Okay. And next class comes bacilli. So bacilli is nothing but bacilli means rod shaped structure. It is derived from the word baculus. Okay. It is derived from the word baculus. Baculus means rod. Baculus means rod. And again, if single rod is present, then it is called by the term monobacilli. The same, if it occur in pair, then it is called by the term diplobacilli. Then if it is occurring in chain, 1 by 1, next to 2, next to 2, next to 2, then bacilli in chains. Okay, streptobacilli. And at last, if it is occurring in cluster, then it is bacilli in clusters. Okay, I have quoted the example: Bacillus cereus, monobacilli, Klebsiella pneumonia, and Streptobacillus moniliformis, Bacillus anthracis under Streptobacilli. Okay, and bacilli in cluster it covers Bacillus anthracis. Okay, so these are the things that is coming under. And another category, Coccobacilli. So Coccobacilli, and again cocci, it represents the oval shape. Okay, so here bacilli represents the rod shape, this spherical and rod, and again it forms a short stumpy shape. Okay, it forms a very short stumpy shape. Here you can able to see the structure of Coccobacillus. Okay, and palisads are nothing but they have bent at the point. While dividing, it bent at a particular point. Here this green color palisads, this is best example is Corynebacterium diphtheriae. Corynebacterium diphtheriae is coming under the category palisad. Okay, so Corynebacterium diphtheriae, it is responsible for causing the disease diphtheria. Okay, and another category, flagellated rods. Example, Salmonella typhi, which is responsible for causing the disease typhoid. Okay, so now another category, spirals. So spirals are nothing but, here we are having 3 categories under spirals. So first one, vibrios. Vibrios are nothing but it is a rod but occurring in a curve. A rod occurring in the form of curve, it is called as vibrios. Okay, comma shape or curve shape rods. Okay, so example, Vibrio cholerae. So Vibrio cholerae responsible for causing the disease cholera. Okay, and another category, spirella. Okay, spirella, it is having a rigid spiral structure. Spirella, and another one, spirochaetes. So spirochetes are flexible body. Okay, helical flexible body, and this spirella are rigid spiral structure. Okay, it is a rigid spiral structure. Spirochetes are flexible spiral structure. Okay, and under spirella I have given the examples, and spirochetes separate examples are there. Okay, so this is the summary chart showing different shapes of bacteria. Okay, and we are moving to the flagella types. Okay, so basically based on the presence of flagella, we can categorize them into flagellated bacteria and non-flagellated bacteria. Okay, so if flagella is absent, then it is called by the term atrichous. So A represents absence, so absence of flagella. Again, the example is Corynebacterium diphtheriae. And coming to flagellated bacteria, monotrichous, lopotrichous, amphitrichous, peritrichous. Mono, only one flagellum is present. Lopo, bunch of flagella or single flagella at one end. Okay, lopo means single end. Okay, either single flagella at one end or a group of flagella at one end. Amphitrichous, amphi means both. Okay, so both end it consists of many different types of bacteria. And then comes the another type, peritrichous. So peritrichous is nothing but the flagella is evenly distributed throughout the surface of the bacteria. Okay, and here you can able to see the picture. Okay, so atrichous, absence of flagella. Monotrichous, single flagella. Then lopotrichous, single-ended flagellum is present, and amphitrichous at both ends. Okay, peritrichous throughout the surface. Okay, and based on the flagella, we can decide either it is a motile bacteria or a non-motile bacteria. Okay, and majority of the gram-negative bacteria is coming under motile bacteria. Okay, maximum all gram-negative bacteria are motile. Okay, few exceptional cases are there. Okay, and here, based on the nature of moving, with the help of this flagella, these are referred as organs of locomotion. They can able to move. Okay, we can categorize them motility into various titles: tumbling motility, gliding motility, stately motility, darting motility, swarming motility, and corkscrew motility. Okay, so these are the different types of motility. And now nutritional requirements. So based on the nutritional requirements, how they are preparing their food, we are categorizing them into two different cadres: autotrophs and heterotrophs. Autotrophs, the term auto is nothing but self. They can prepare their own food. Okay, whereas in case of heterotrophs, they depend upon other organism to prepare their food. Okay, again, these autotrophs are categorized into photoautotrophs and chemoautotrophs. Photoautotrophs are nothing but they can prepare their own food with the help of energy source, light. Okay, light as the energy source. Here, if they utilize inorganic compound as the energy source, then they are called by the term chemoautotrophs. Okay, if you utilize light, then it is photoautotrophs. If they utilize inorganic compound, then chemoautotrophs. Okay, and another one, organic compounds using carbon source, heterotrophs. If they utilize light to prepare their ready-made foods, then they are called as photoheterotrophs. Okay, if they utilize organic compounds as their energy source, then they are called by the term chemoheterotrophs. Okay, so this is about based on the nutrition. And based on oxygen requirement, we are categorizing the microbes into 5 main titles. Okay, so first one, obligate aerobes, obligate anaerobes, facultative aerobes, aerophiles, microaerophiles. Okay, so under obligate aerobes, so what is obligate aerobes? They require oxygen for their growth. Okay, so only if oxygen is present, they can able to grow. Okay, they can able to survive or they can able to grow only in presence of oxygen. Those categories referred as obligate aerobes. And another one, obligate anaerobes. Obligate anaerobes, they do not use oxygen for their growth. Okay, they depend primarily on other process for their growth, that is fermentation, anaerobic respiration, bacterial photosynthesis, and methanogenesis. Okay, these are all the various route through which they can require energy. Okay, and presence of oxygen kills or inhibits their growth. Okay, obligate anaerobes. Another category is there, because another category, that is aerotolerant anaerobes. Aerotolerant anaerobes also follows anaerobic type of respiration. But the difference between aerotolerant and obligate anaerobe is, obligate anaerobe cannot able to survive in presence of oxygen. Okay, if oxygen is present, either it inhibits or kills their growth. Those categories are called as obligate anaerobes. Okay, whereas aerotolerance, they can tolerate the presence of oxygen. Even though oxygen is present, the aerotolerance never bother about the presence of oxygen. They continue grow, but they do not utilize oxygen. They continue their growth by utilizing the fermentation pathway. Okay, so this is the major difference between obligate anaerobes and aerotolerance. And the third category, facultative anaerobes. Facultative anaerobes are also called as facultative aerobes. Facultative aerobes or facultative anaerobes. Okay, so here they switch between this aerobic and anaerobic respiration condition. Either they can utilize based on their convenience, either they can go with aerobic respiration or they can go with anaerobic respiration. And the last one contains microaerophiles. Microaerophiles require oxygen, but it requires oxygen at a very minimal level, only 0.2 atmospheric pressure. Okay, so based on temperature requirement, we can categorize this into mesophiles, thermophiles, extreme thermophiles, and psychrophiles, acychrophile. Okay, so mesophile, that are the organisms that can grow at the normal temperature, that is 37 degrees Celsius. So 37 degrees Celsius, that is our body temperature. Okay, if a microorganism can able to grow or able to withstand at this particular temperature, then those categories are referred as mesophiles. Okay, example, E. coli, Mycobacterium tuberculosis, Pseudomonas aeruginosa, Streptococcus pyogenes. Okay, then comes another category, thermophiles. Thermophiles, they can grow between the temperature of 45 to 70 degrees. Okay, thermophiles can grow between the temperature of 45 to 70 degrees. Okay, then next one, extreme thermophile, they can able to withstand somewhat higher temperature, which is 115 degree, 80 degree to 115 degree. They are also referred as hyperthermophiles. Okay, hyper means above. So above the thermophilic condition, they can able to withstand. So now comes another category, psychrophiles. So psychrophiles, they can able to grow even at 0 degrees Celsius. Okay, so sometimes we are storing our food under refrigerated condition. Even at refrigerated condition, some food items or some eatables get spoiled. Okay, so that is mainly because of this psychrophiles. Okay, and again, psychrophile, another variant is called as apsychotroph. Okay, a psychotroph that can able to grow between the temperature of 10 to 15 degrees Celsius. Okay, so this is classification or nutritional requirement based on the temperature. Then comes the effect of pH. So based on the pH, we can define 3 cardial points. That is 3 important points. 3 important points for defining the growth of the bacteria or defining the growth of the microorganism. What are all the 3 important points? The first comes minimum pH, optimum pH, and maximum pH. Okay, so this minimum pH is nothing but below which it cannot grow. Minimum pH is the pH below which it cannot able to represent any growth. Okay, optimum pH is the very best pH at which the bacteria or any microbe can able to grow at its maximum. Okay, and maximum pH, above the pH it cannot able to grow. Okay, you should not confuse with the term maximum. Maximum, it is not representing maximum growth. Okay, minimum pH, maximum pH, and these two are the 2 endpoints. Okay, below minimum pH there is no growth, above maximum pH there is no growth. Okay, so starting from minimum pH it starts growing. okay, and it stops its growth when it reaches the maximum pH, okay. And here you got a center point or average point is defined by the term optimum pH, at which you can able to see the major growth or the best growth of the microorganism, okay, best growth of the microorganism. And these 3 points are very important: minimum pH, optimum pH, and maximum pH. And here I have given few examples. For Staphylococcus, the minimum pH is 4.2, maximum pH is 9.3. And for optimum pH, that is between pH 7 and 7.5, the Staphylococcus aureus can able to show the best growth, okay. So now comes the another 3 important pH or another 3 points based on the pH: acidophiles, neutrophiles, and alkaliphiles. So acidophiles are nothing but it can able to grow at an optimum pH or acidic pH, okay. Acidophiles are the microbes that can able to grow below neutral pH, okay. So in the graph you can see the acidophiles can able to show what is the growth rate of acidophiles at this particular points, okay. And neutrophils can able to grow best at the neutral pH, okay. And alkaliphiles, they can show best growth at the alkaline conditions, okay. So under pH, 3 cardinal points: minimum pH, maximum pH, optimum pH, okay. And 3 category under based on the pHs: acidophiles, neutrophiles, alkaliphiles, okay. And effect of osmotic pressure, and based on the effect of osmotic pressure we can categorize them as, or 2 major terms to be known as plasmolysis and plasmoptysis. So plasmolysis is nothing but sudden exposure to hypertonic solution, okay. So if the microorganisms are transferred to hypertonic solution, that is highly concentrated solution, what happen? There is a osmotic withdrawal of water, okay. So if it is transferred to concentrated solution, there occurs withdrawal of water, okay. So the cellular contents are withdrawal and shrinkage of protoplasm occur. So this shrinkage majorly noticed in case of gram-negative bacteria, okay. If it is transferred from, if a bacteria is transferred from dilute solution to concentrated solution, then you can able to see the process of plasmolysis, okay. So plasmolysis is accompanied by shrinkage of water, okay, shrinkage of water. Okay, so now comes the another term, plasmoptysis. So plasmoptysis is nothing but the sudden transfer from a concentrated solution to a dilute solution. So, or it may be a distilled water, okay. From concentrated solution, if the microorganisms are transferred to a distilled water or any other solvent, then it causes plasmoptysis. Plasmoptysis is nothing but imbibition, okay. It absorbs more amount of solvent and swelling, and it finally leads to rupture of the cell, okay. And then comes water availability. So water availability is nothing but the pure water is represented as water availability, and it is represented by the symbol AW. So the AW value of pure water is 1. That is 100% solvent molecules, okay. If the pure water is affected by the addition of solutes, okay, by the addition of either salt or sugar, the pure water value gets decreased, okay. The value gets decreased, and because of this decrease in value, the water availability point or the water availability ratio is also decreased. The water availability ratio is also decreased, okay. So here the microorganism can able to live over a range of 1 to 0.7. If the water availability ratio is still reduced below 0.7, then the microorganism growth rate is also decreased, okay. So this is the thing related with nutritional requirement. And at last, to end with, we can able to see the availability of salt ratio. So what is the proportion of salt present here? So here various proportions of salt is present. So salt, it includes the term sodium chloride, So if it requires some sodium chloride, some amount of sodium chloride for their growth, then they are referred by the term halophiles. Okay, so halophiles, it is also referred by the term osmophiles. And second one, mild halophiles, it requires 1 to 6% of salt. So if it requires 1 to 6% of salt, then it is called by the term mild halophiles. If it requires 6 to 15% of salt, if it requires 6 to 15% of salt, then it is called by the term moderate halophiles. Okay, more than 15% of sodium chloride it requires, then it comes under the category extreme halophiles. What it is? Extreme halophiles. Then sometimes they can able to grow even at moderate salt concentration. Okay, so if it grows at moderate salt concentration, then they are called by the term osmotolerance or halotolerance. Okay, and the last one, xerophiles, these are the organisms that can able to grow in dry environment, that is lack of water, promote the growth of microbes. Okay, so based on salt, so these are the various categories. Okay, and comes the last two important terms, symbiosis and antagonism. So symbiosis is nothing but the term sym, which is nothing but summation of activity. So growth of one microorganism favors the growth of another microorganism, then it is called by the term symbiosis. So here the example is Staphylococcus aureus and Haemophilus influenza. Okay, so both can support the growth of one another, then it is called by the term symbiosis. And another one, antagonism. Antagonism, and again anti means against, so the growth of one organism is determinantal, okay, is counter to the another organism. Okay, so here Pseudomonas controls the growth of gonococci. So these are all the various terms related with the bacteria structure, bacteria classification, nutritional requirements. Okay, so next we are moving back to another slide. Sir, shall I take a few more minutes, sir? I can end with biochemical test.
Yes, ma'am.
Okay. So I hope the slides are visible. Yes, in-back slides.
Yes, ma'am. Yes, ma'am. It's clear.
Okay, thank you, sir. Thank you. So since the time is very less, just I would like to give a brief about in-back test. Okay, so here comes the biochemical test. So biochemical characters are very much important to identify or to separate the bacterial cells. Okay, so two category of biochemical tests are present. So based on the based on the biology of the microorganisms, we can categorize them into two categories: extracellular enzymes and intracellular enzyme. Okay, so this extracellular enzyme, based on the enzymes which produces are secreted outside the cell. Okay, so we can have four different biochemical tests: starch, gelatin, lipid, and casein. Okay, and intracellular enzymes, these enzymes are present, are secreted inside the cellular cavity. Okay, so this is also referred as endozymes or endoenzymes. Okay, intracellular enzyme. So here again I have given the list of tests. The very important thing which is coming under GPAT syllabus is sugar fermentation test or carbohydrate fermentation, and another one is IMVAC test. Okay, so now I will discuss with you about IMVAC test. So this IMVAC test is mainly used to differentiate the gram-negative intestinal bacteria, which belongs to the family Enterobacteriaceae. Okay, it is used to differentiate gram-negative intestinal bacteria, belongs to the family Enterobacteriaceae, particularly Escherichia coli and Enterobacter group. Okay, Enterobacter and also the Klebsiella group. Okay, so here based on their biochemical property, So biochemical property is nothing but based on the number of enzymes or the type of enzyme they are producing, the action, the followed biological action is also different. Okay, so here the term IMVAC is abbreviated as follows. I represents indole test, M represent methyl red test, V represent Voges-Proskauer test, and C represents citrate test. Okay, so here this in between one small letter I, which is used to represent in. Okay, that is here we are utilizing citrate medium, and inside the citrate medium we are adding this microorganism. Okay, so that's why we have got the additional letter I, but here we have only 4 biochemical tests. Okay, so indole test, methyl red test, Voges-Proskauer test, and citrate test. Okay, so I think directly we can move to the summary chart, so that I will explain. Okay, so here in case of indole test, wait a minute. Okay, so this is the summary chart to indicate the IMVAC test. So under indole test, here we are using the SIM broth. Okay, here we are using the SIM broth, and this is the test which is used to identify the biochemical characteristic feature. Okay, the biochemical characteristic feature of the particular or the given microorganism. Here we can identify what type of microorganism is present. Okay, so here we are using the COVAX reagent to identify the reaction. And in case of methyl red test, we are using methyl red Voges-Proskauer broth, and the indicator we have used is methyl red indicator. And Voges-Proskauer test, the same broth we are using, but the identification reagent is Barrett's reagent. And the last one, citrate utilization test, we are using Simon citrate broth, and the indicator bromothymol blue, which is present inside the same media. Okay, bromothymol blue is present inside the same medium. Okay, so to get deep into the test. Okay, so in case of indole test, so it is mainly used to identify a particular property, either it contains, either it is having the ability to degrade the amino acid tryptophan. Okay, so here we can able to check whether the tryptophan amino acid is degraded by the given microorganism or not. Okay, so again, this tryptophan is degraded by the enzyme tryptophanase. This tryptophan is degraded by the enzyme tryptophanase. So if tryptophanase is secreted by the microorganism, then it will degrade the tryptophan, and the resultant product is identified with the help of identifying agent. Okay, so in all the biochemical tests, we supposed to prepare a particular media. Okay, to perform whatever type of biochemical test, we need to prepare the culture media for the respective test. Okay, each media we supposed to use a different culture media. So what is the required culture media that we have to prepare? So after preparing the media, we supposed to inoculate the microorganism, and the third step is incubation. After incubation, we have to identify the growth. We can visualize the growth, and by seeing the growth, we can identify the microbes by means of adding the identification reagent. Okay, so initially we are preparing the medium, preparation of culture medium, then second step, inoculation of microorganism, and third step, incubation of the medium, and the last one, identification. Okay, so here the difference comes what media we are using, and according to the media, what is the identification test we are conducting. Okay, so in first indole test, we are going to identify the ability of the microorganism to degrade tryptophan. Okay, so for degrading tryptophan, the microorganism supposed to produce tryptophanase. Okay, so if the microorganism produce tryptophanase, it will degrade the tryptophan. And based on the degradation of tryptophan, or the tryptophan is not degraded, we can able to identify the ability of microbe. Okay, so after incubation, if the tryptophan is degraded or not degraded, using that we can conclude what is the result. Okay, so here the tryptophan, how it is degraded. Okay, so this tryptophan, by means of degradation, it produces indole, pyruvic acid, and ammonia. So this indole moiety, that is the degradation product, reacts with our identification reagent. What is the identification reagent? COVAX reagent. Okay, so COVAX reagent is chemically called by the term para-dimethyl aminobenzaldehyde. Para-dimethyl aminobenzaldehyde. So it reacts with COVAX reagent and it produces quinone-diol red violet colored compound. Okay, so you can able to see this is the appearance of cherry red color. Okay, so if this color present, if this color appears, then it indicates indole positive reaction. Okay, indole positive reaction. So indole positive reaction is positive, or it indicates the presence of E. coli, and indole negative reaction indicates the presence of Enterobacter aerogenes. Okay. So then moving on to methyl red test. So methyl red test, here we are identifying the ability to oxidize glucose. Okay, so if glucose is oxidized, okay, if glucose is oxidized, it can produce various end products. It can produce various end products. So by the oxidation of glucose, it will produce lactic acid, acetic acid, and formic acid. It will produce lactic acid, acetic acid, and formic acid. So under acidic pH, the methyl red is red in color. Acidic pH, it is red in color. Whereas under alkaline pH, okay, under alkaline pH, it becomes yellow color. So how this alkaline pH come? This acidic end products again reacts together to produce acetyl methyl carbinol. To produce acetyl methyl carbinol in case of Enterobacter aerogenes. Okay, in case of Enterobacter aerogen. Okay, so if it is E. coli, it maintains basic pH and the methyl red is red in color. In case of Enterobacter aerogen, basic pH and it is in yellow color. Okay, so then comes the next test. After methyl red, the next one is Voges-Proskauer test. So here you can see the methyl red test, red color and yellow color. Okay, so Voges-Proskauer test, and again we can tell it is a confirmatory test to differentiate E. coli and Enterobacter aerogen. Here instead of methyl red, we are using the Barrett's reagent. We are using the Barrett's reagent, and here the media what we are using is same. Methyl red Voges-Proskauer broth, the same media we are using, but the reagent what we have used is Barrett's reagent. It contains a mixture of alcoholic alpha naphthol and 40% potassium hydroxide solution. So here what happens, the end product supposed to produce a different color in case of Voges-Proskauer test. Okay, so you can get a better idea by seeing this picture. So in case of Voges-Proskauer, the color development indicates what is the difference between the E. coli and the Enterobacter aerogenes. Okay, so finally comes citrate utilization test. Here we are using the Simon citrate medium. So Simon citrate medium, here we are using citrate as the major source for the microorganism. If the microorganism want to utilize this citrate, then it should contain citrase enzyme. Okay, so if citrase enzyme is present, it utilize the citrate moiety present in the given media. Okay, then correspondingly we can able to get the color change. Okay, so here to identify the acidic or basic nature, bromothymol blue is included in the media itself. So bromothymol blue, it is one among the ingredient in the culture media, which is used as an indicator to indicate whether the media is in acidic pH or basic pH. Okay, so if green color, the media represents acidic. If the media is alkaline, then it got blue color. Okay, so based on this reaction, if citrase enzyme is present, then the color changes to green. Okay, if citrase enzyme is produced by the microorganism, the reaction occurs in this sequence and finally we get blue color. Okay, if citrase enzyme is not present, then we conclude there is no color change. It represents the presence of E. coli. Okay, so this is the final summary chart. And what are all the species which shows positive and negative reactions with this indole, methyl red, Voges-Proskauer and citrate utilization test. Okay, so I hope I shared little things regarding the microbiology to just overview with the provided time limits, and sorry to take more than the allotted time, more than 60 minutes I took. So sorry for time delay. So I hope the participants got few information from my presentation. So I would like to thank everyone for this opportunity. So thank you very much.
Thank you, Doctor. Thank you so much, Doctor, for the elaborate and the slide presentation. Thank you, ma'am. Thank you, ma'am.
Yes,
Thank
yes,
you, ma'am.
Sumathy madam, thank you for your extensive talk. Your slides are very nice. The students
Thank you,
also
madam.
gave
Thank
the feedback. Even
Thank
though basic only given, it's a very nice presentation. Elaborately covered all the points of the basic things. Sumathy madam, thanks
Thank you,
for
madam.
your
Thank
effort
you.
and service to the GPAT class.
Thank you, madam. Thank you very
Thank
much.
you, ma'am. Jaykumari, ma'am, you can take over.
Yes,
for the
yes,
next
sir.
session.
Yes, sir. Yes, sir.
Please
Yes,
go ahead,
students, good morning. Now the second session is going to start. The second session speaker is a well experienced in pharmacology, Dr. V.K. Kalaiselvan, sir. I am very happy and very pleasure to introduce my friend and colleague, long-term friend, Dr. V.K. Kalaiselvan, sir. Actually, presently he is working as a head and department of pharmacology at Annamalai University. Before a professor in working in Annamalai, he was worked as a principal, former principal in KK College of Pharmacy. At that time, I was very close with him. He was a well-known personality in pharmacology and other activities also. He completed her UG B.Pharm degree from Annamalai University and PG from MGR University. He completed PhD degree in Annamalai University. He has a total teaching experience of more than 47 years. Under that one, he have more than 26 years. Under that, he have a research experience 15 years. He has a recipient of many awards also. He has published more than 50 numbers of publication, both national and international. He guided about 26 B.Pharm projects and 20 M.Pharm projects, guided about 5 PhD program, and he is guiding 5 numbers of PhD students. He has a recipient of many funding projects also. Receipt of the UGC major project, which is worth about rupees 9.5 lakhs. He is also going on a project, undertaking a project. The project is going on under his supervising. Interdepartment RUSA project and also interdepartment project, which is worth about 38 lakhs in Annamalai University. He has also expertise and experience in appointing various examiner as an external examiner in various universities, more than about 7 universities. He was and also he was appointed as a question paper setter and PhD evaluator in many universities as a PhD evaluator also. With this short profile, I welcome Dr. V.K. Kalaiselvan, sir, for his sharing his valuable information and experience, expertise, knowledge in pharmacology. Mainly the topic is relevant to the students. I request all the students to please attend this session, is very useful to you. Given that podium to Uma Maheshwari, madam. Thank you, madam.
Thank you, Doctor. Dr. Jaykumar, thank you.
Yes, yes, madam. Yes, madam.
I came to know only now that you worked in Dr. Kalaiselvan previously, ma'am. Thank you.
Actually, we both are very close friends, madam.
But
He was worked in KK College of Pharmacy.
And last year's also was very good, ma'am. Thank you. You have taught many people. Thank you.
சார் ஒரு 2 minutes சார் ஜாயின் பண்ணிட்டு இருக்காரு.
சரி சரி, நோ ப்ராப்ளம். ஸ்டூடெண்ட்ஸ் நிறைய இருக்காங்க இன்னைக்கு, பரவாயில்ல. ஸ்டூடெண்ட்ஸ் ஆல்சோ ஐ ரெக்வெஸ்ட் ஆல் தி ஸ்டூடெண்ட்ஸ், ப்ளீஸ் ஆஸ்க் யுவர் friends to join that one, because today's topic is very important. ANS system, autonomic peripheral autonomic nervous system. There are many questions in GPAT they are asking. So once if you know this type of basic pharmacology, then only you can answer for the drug action and drug mechanism of action and everything. So I request all the participants, please you message to your friends also to ask them to immediately join. immediate accept
சொல்லுங்க madam.
பண்ணிட்டீங்க. I thank to you.
Yes, thank you madam. Thank you madam. Thank you. Shall we start now? Start
You
பண்ணவா?
start, sir. Yes, sir.
Right, right. Right. Good morning, one and all. I thank to IPGA team, particularly GPAT team under the head of Dr. Madam Jaikumari Madam and Meenakshi Madam, Uma Madam, and other team work. Very nice work what is going on. Last year also very. Then the output was outstanding output what we heard from so many colleges. And the students also they are benefited. I thank to one and all. Good morning, dear students. This is my topic is autonomic nervous system and related the drugs, cholinergic drug, cholinergic blockers, adrenergic drug, adrenergic blockers. This is my name is Kalaiselvan. Already they introduced. Next one, general introduction about the particular topic. Autonomic nervous system, they are actually the nervous system divided into two heading, central nervous system, peripheral nervous system. Under the peripheral nervous system, again subdivided into autonomic nervous system. Another one is somatic nervous system. Under the heading, general consideration, then cholinergic nervous system and drugs, and anticholinesterase and its drug. Adrenergic, what are the cholinergic transmitter, adrenergic transmitters, then what are the receptors are involved in the both nervous, cholinergic and adrenergic system. What are the agonist drugs are there, antagonist drugs are there. This was everything already you know. Just the introduction what you are given, what you discussed. Here, the diagram what it shows peripheral, central, the peripheral nervous system under that only afferent division under that. Autonomic nervous system divided into subdivided into parasympathetic and sympathetic, and afferent division somatic nervous system is a single nerve, absence of ganglion, directly is maybe the nerve end and neuromuscular junction, NMJ. And the NMJ one receptor is named as nicotinic muscular receptor. Again, the autonomic nervous system, parasympathetic nervous system and sympathetic nervous system, the outline sketch. There is section myelinated and non-myelinated. I mean pre-ganglionic parasympathetic nervous system. Another one is post-ganglionic parasympathetic nervous system. The pre-ganglionic and post-ganglion nervous system in between, that is ganglion is there. In the ganglion or receptor is there. That name of the receptor is nicotinic receptor. Nicotinic receptor is subdivided into NN, nicotinic neural receptor. I mean particularly present in ganglion and adrenal medulla. Another one more subdivision is NM receptor, that is present in somatic nervous neuromuscular junction. Agonist and antagonist is there. Sympathetic nervous system, preganglionic sympathetic nerve, postganglionic sympathetic nerve. In between the nerve, there is ganglion is there. There is also NN receptor is there, ganglion. Cholinergic nervous system, I mean parasympathetic nerve ending, neuroeffector junction. I'm talking about parasympathetic nerve ending, neuroeffector junction. That junction receptor is the name of the receptor is muscarinic receptor. The muscarinic receptor is divided into 5 majorly, M1, M2, M3. There is totally 3 receptors. Again, M4 and M5s are autoreceptor. Next one is sympathetic nervous system, preganglionic sympathetic nerve, postganglionic sympathetic nerve. Again, the preganglionic and postganglionic neurotransmitter is noradrenaline. Preganglionic parasympathetic and postganglionic parasympathetic are acetylcholine is a neurotransmitter. Preganglionic sympathetic nerve is acetylcholine is a neurotransmitter. Sympathetic nerve ending neuroeffector junction, that is the receptor is named as alpha major, minor, beta receptor. Another one more nerve is there, is nothing but adrenal medulla. From the adrenal medulla, adrenal gland again divided into adrenal medulla, adrenal cortex. From the adrenal medulla, release of hormone as well as a neurotransmitter, adrenaline. These adrenaline is a single nerve. This single nerve release adrenaline, carried adrenaline, the nerve ending neuroeffector junction, release of adrenaline bound to alpha and beta receptor. So totally, the nervous system totally which is having major is 3 receptor, major nicotinic receptor, which is present in ganglion and adrenal medulla. Next place is muscarinic receptor present in postganglionic parasympathetic nerve and the effector organ, muscarinic receptor. Next one is sympathetic neuroeffector junction, alpha receptor is there. Adrenal medulla release adrenaline, adrenaline which is having alpha and as well as beta receptor action. These are the major thing. Somatic nerve already we are discussed. Because we have a short time, that's why what I go for somewhat fast. Please listen, dear students. Then what about the functions? Physiological function. The physiological function of sympathetic division increased force of contraction and rate of contraction, mainly the heart. Increase the rate of contraction and force of contraction. The blood flow also may be increased to the skeletal muscle and heart stimulation. They have a fly and fight mechanism. Fly and fight mechanism. The adrenaline, whenever our body demand is a requirement is at that time is maybe fly, again is maybe fight. Compensate the loss of your body energy as well as memory loss and other things only temporary. So immediately maybe the fly and fight mechanism, what it is adrenaline. Next one, the functions of parasympathetic nervous system, rest and digest. The role of CNS action and the motor nervous system, the sensory input and as well as peripheral structure, provide the information about all those things. Reflex arc, what it is says, afferent impulse and then there may be efferent impulse. The received message again directly goes to the the nerve ending as well as the effector junction. The receptor is bounded, again is maybe either maybe executory action or maybe inhibitory mechanism, depending upon the role of the one thing. Maximum parasympathetic nervous system, execute responses. The sympathetic nervous system, inhibit responses except intestine. Parasympathetic nervous system, all the responses are except escape action, except what you call heart. Next one, emotional autonomic nervous system. This activities may be modified with stimuli and evoke the feelings and range of the fear and pleasure. Chemical signaling, just now we have discussed. The neurotransmitter, totally, acetylcholine one neurotransmitter, noradrenaline one neurotransmitter, and adrenaline one transmitter. These are all neurohumoral transmitter in all peripheral nervous system. Action may be different. One by one is maybe action may be. There is one more hormone is that adrenaline, which act like a 2-in-1 action. One side is hormone, other side is neurotransmitter. These are all already discussed. Impulse formation and conduction depending on the influx of calcium ion. Then there is depolarization, repolarization, influx of sodium and influx of calcium, efflux of potassium, there is a depolarization. Next one is efflux of calcium followed by sodium and influx of potassium, that is repolarization. Depolarization is nothing but execute response or maybe stimulation or contraction. Next one is repolarization, relaxation or maybe inhibitory mechanism. These are all things are covered in the thing. The transmitter already we discussed. Preganglionic parasympathetic nerve, acetylcholine is a transmitter. Postganglionic nerve, there is acetylcholine is a transmitter. Preganglionic sympathetic nerve, acetylcholine is a neurotransmitter. Postganglion sympathetic nerve, noradrenaline is a transmitter. Then adrenal medulla, adrenaline is a transmitter. Then somatic nerve, acetylcholine is a neurotransmitter. These are the things, what are the different mechanisms taken place, how it is synthesized, how it is maybe released. Number one, there is maybe transmitter, is synthesized and stored in the vesicle. Second one, an action potential involved presynaptic terminal. Third one is depolarization of the presynaptic terminal causes opening of the gated immune calcium channel. Then the influx of calcium is enter. Then fifth one is the calcium causes vesicle, diffuse the presynaptic membrane. Sixth one is the transmit and release the synaptic cloud. Here this area. Next one, 7, transmit and bind the receptor. Here neuroeffector junction cholinergic nervous system in muscarinic receptor. Adrenergic nervous system mean alpha receptor. Neuroeffector junction adrenal end mean alpha and beta receptor mechanism. Then after that, it is going to metabolize. After the giving the suitable physiological reaction, that is metabolized in the presence of cholinesterase enzyme. These cholinesterase enzyme is divided into two, pseudocholinesterase enzyme and true cholinesterase enzyme. Again, remaining are, that is actually a reuptake mechanism is there. The reuptake is not metabolized, that is retrieval the vesicle membrane from plasma membrane. Again, further is transmitter synthesized, then stored in the vesicle. Again, maybe this is cycle, one of the cycle. Next one. This is actually basic for your peripheral nervous system, and that is nothing but parasympathetic and sympathetic outflow from your spinal cord. There is also nicotine receptor is there. Stimulation and release of preganglionic fiber and postganglionic fiber, both parasympathetic and sympathetic. Here, craniosacral outflow. Craniosacral outflow. That is the craniosacral outflow. Parasympathetic nervous system is involved. Then it is excitor and as well as inhibit respond. The main place is eye, lacrimal gland, salivary gland, heart, lungs, upper GIT tract, lower GIT tract, bladder, and gentian organ. These all the things are covered by parasympathetic nerve. Outflow is craniosacral outflow. Next, sympathetic nervous system, thoracolumbar outflow. Thoracic nerve and lumbar nerve. There is from there only is innervation, presynaptic and as well as postsynaptic. Presynaptic is sympathetic nerve and postganglion is sympathetic nerve. In between the ganglion is there. What are the functions are there? Where the physiological actions are there? Mean head and neck, eye, that is maybe dilatation, blood vessel dilatation, salivary gland, there will be a thick salivary secretion, heart may be increase in rate of force of contraction, lungs relaxation, adrenal medulla release of adrenaline, then liver, GIT tract, gallbladder, gentian organ, blood vessel, sweat glands, and etc. This are all the flow chart of your nervous system. General consideration already we have discussed. Then we go for what you call these are the things already I point out. Synthesize and release of acetylcholine. That is stored, again released, binding to suitable receptor. I mean, that is the cholinergic receptor is 2, nicotine receptor and muscarinic receptor. Nicotine already pointed out, NN and NM receptor. Again, muscarinic receptor is 5 receptor. Then finally it may be metabolized. Again, these are the how it is synthesized. Adenosine triphosphate and coenzyme in the presence of acetate activating reaction gives acetyl coenzymes, then cholinesterase, finally form acetylcholine. This acetylcholine is gives so bind to suitable receptor, either nicotine receptor nor muscarinic receptor. Finally, is going to metabolize. This metabolic product is again converted into acetylcholine and choline, acetic acid and choline. Again, the choline is again recycled, again reuptake, again is reformed. Acetylcholine. Is a storage deposit already the flow chart already discussed. These are the two enzymes. I mean, the pseudocholinesterase, true cholinesterase enzyme. The recycling choline only just now we are discussed. This is also already I was pointed out. Synthesis of acetylcholine, what's the role behind? Transport to the choline and inhibited by hemicholine. And this is the drug name. Uptake and storage vesicle. Acetylcholine is to protect from the degradation of the second point. A third point, a release of neurotransmitter, then bind to, fourth one is bind to suitable receptor. Fifth point, degradation of the acetylcholine, rapidly hydrolyzed, converted in the presence of acetylcholine esterase. There is one more classification is there. They are going to inhibit the cholinesterase enzyme. Acetylcholine is not metabolized. This is further divided into reversible and irreversible. That is two, enzymes here, reversible and recycling, and reuptake. Choline again taken up the neuron. These are the receptor already discussed. M1 receptor. I pointed out some other things located in ganglion cell and central neurons. It plays a major role of GIT secretion, relaxation of esophageal sphincter muscles, and vagal stimulation. M2 specifically heart. Specifically present in heart, but a negative feedback mechanism, negative inhibitor mechanism decreased force of contraction, a decreased rate of contraction. It's M2 specific action. M3 receptor directly present in visceral smooth muscle contraction, glandular secretion. Mainly they pointed out M3. Okay. Next one, NM receptor. NM receptor already I pointed out. NM receptor where it is present in ganglion as well as adrenal medulla. Parasympathetic ganglion, sympathetic ganglion, nicotinic neuronal receptor. Nicotinic neuronal. This is stimulated by dimethylphenyl fibranism. That is the stimulant agonist. NM receptor present in skeletal muscle, mainly neuromuscular junction. This is mediated to skeletal muscle. It's a single nerve, absence of ganglion. There is no ganglion is there. This is selectively stimulated phenyl trimethyl ammonium. Blocker is D-dibochlorine and curare and alkaloids. D-dibochlorine, galamine, pencuronium, alcuronium. These are all blockers. These are all peripheral skeletal muscle relaxant drug. Peripheral skeletal muscle relaxant drug. Other name is neuromuscular blocking agent. This is also used for pre-anesthetic medication, major surgery. Major surgery is used for pre-anesthetic medication. Okay, next one. So these are all straight I go for pharmacology of this one. Classification. Major one, any examination, definitely the classification is there. The classification according to the basic structure. Cholinesterase, cholinester. The cholinester, acetylcholine, methacholine, methanocol, carbachol, and methanocol. These are all cholinester basic nucleus. Alkaloids, they are obtained from mushroom, muscarine, acridine, pilocarpine. And synthetic compounds, oxotermetrine and metacloramide. Acetylcholine, mechanism action and pharmacological action. These transmitter already we are discussed pre and post ganglionic parasympathetic nerve and pre-ganglionic sympathetic nerve. There is acetylcholine is present. And one more area, somatic nerve. Somatic nerve also may be present. Muscarinic action, just now we discussed. Negative mechanism, inhibit mechanism. Directly acting on the hyperpolarized SA nodal, decreased diastolic depolarization. Then it is induced bradycardia, decreased heart rate, decreased force of contraction. Refractive prejudice also may be increased, decreased cardiac output. All those things are negative. Smooth muscles are contraction. All the smooth muscles are contraction. Blood vessels are always dilated. Endothelial dependent relaxation factors may be dilated. Glands, all the glandular secretions are increased. All the glandular secretions are increased. For example, salivary gland, sweat gland, lacrimal gland, mucus secretion, bronchial secretion, all the things are increased. In eye, that's, the eye that is actually meiotic effect is there, directly bound to circular muscle that it is produce meiotic effect. Contraction of a circular muscle is iris, is meiosis, ciliary muscle. This is mainly used for glaucoma, not directly acetylcholine, but that is a transmitter. It may be induced in normal secretion and meiotic effect is there. Then the sympathetic nervous system alpha receptors are dilated. Both are vice versa. Modulator effect. Nicotinic action, autonomic ganglia, parasympathetic ganglia and sympathetic ganglia stimulated. There is a release of acetylcholine, another side noradrenaline. Skeletal muscle, somatic nervous system. Somatic nerve bound to nicotinic muscular receptor, contraction of skeletal muscle. The adrenal medulla, release of adrenaline. That is produce a tachycardia, increased force of contraction and rate of contraction of cardiac muscles, the cardiac chamber. I is a meiosis, what you call pilocarpine. I'm talking about pilocarpine. CVS pilocarpine action, pilocarpine is opposite effect. There directly acetylcholine bradycardia. Pilocarpine produces a tachycardia in high dose, but a low dose may be fall in BP. Meiosis, I in meiosis, glands or secretion is increased, dose 0.5 to 4% in eye drops. This is a mushroom. In the mushroom only what you are obtained from your, what you call muscarine. Anticholinesterase. The cholinesterase enzyme divided into true cholinesterase and pseudocholinesterase enzyme. Again, what's the role behind under the classification of cholinergic drug? Cholinergic drug, this is one of the classification. What's the reason behind reversible mean? This is only temporary action, again reverse back. Irreversibles are once bounded, then it's not reverse back. Irreversible drugs are poisons, purely poison. This mechanism, reversible some of the example. So I'll give the some of the example. They protect acetylcholine hydrolysis, produce cholinergic effect. In vivo studies potentiate acetylcholine both in vivo and in vitro. Some have additional direct and cholinergic receptor. Reversible drugs, physostigmine. A synthetic drug, neostigmine, predostigmine, ambronium, decamacrine, entropinium, rivastigmine. These are all reversible mechanisms. Irreversibles are once binded is not reverse back. Mechanism action of this one, the reversible cholinesterase enzyme. The acetylcholine system is structural similarities are both anion and ester side. The cholinesterase, reversibles are bound to both anionic side and as well as cation, ester side. But to the short duration may be again reversible because only what it is called reversible cholinesterase enzyme inhibitor. Irreversible cholinesterase enzymes or enzyme, then the drugs are directly bound to only ester side only, not reverse back. These are all poison. Malathion, parathion, TIC20. These are all those who are attempt for the suicide attempt their maximum they are using phosphorus related poison. The phosphorus related poisons materials are irreversible cholinesterase enzyme inhibitor. Clinical symptoms, all the glandular secretions are increased, all the smooth muscle contraction is increased, then heart muscle, cardiac muscle, then cardiac output initially increased, later on cardiac decrease. Negative mechanism, slowly maybe heart rate is decreased, slowly what you call force of contraction is decreased, then what you call intercostal muscle is paralysis, finally death. These are all poison report. The top choice we will see one by one. Irreversible, reversible physostigmine, this is obtained from Calabra bean, is a dried ripe seed, African woody climber, Physostigma and Ruizia. Chemical basic structure is salicylic acid and derivative therapeutic action, anticholinergic antidote. Atropine poison is a very good backup choice, is physostigmine. Next one, mechanism action, just now we discussed. Then these drugs are directly bound to cholinesterase, anionic side and ester side. This time we discussed. Irreversibles are bound to ester side only. Adverse reaction. These are all adverse reaction induced by high dose of cholinesterase enzyme inhibitor, maybe reversible enzyme inhibitor, or maybe poison. CNS, continuous stimulation, fatigue, weakness, bradycardia, palpitation, ENT, increases salivation, lacrimation, myiasis, GI tract, abdominal pain, diarrhea, nausea, then urinary urgency. Next one is muscle twisting, respiration bronchospasm, dysphonia, skin diaphoresis. Continuous sweating is there. So things are there. IV route contraindication for mainly for these drugs are asthma, cardiovascular disease, diabetic mellitus, ganglion, JIT and GU, obstruction, hypersensitivity to physostigmine compounds. IV route, these are the doses. Plasma off-life period 3 to 8 minutes IV. Then peak level 5 minutes. Then duration 30 to 60 minutes. Indication of storage. The contracted toxic anticholinergic effect, anticholinergic syndrome, then is contract with this one. 0.5 to 2 milligram, not more than 1 milligram. Repeated dose 20 to 30 minutes. Atropic poison is mainly used for meiotic treatment of glaucoma. Neostigmine is similar to that of neostigmine. Physostigmine activity. This is bound to both the anionic and ester side. It helps to it helps to prevent relief of urine retention, increase the distance, muscles, tone, bladder, causing bladder contraction, prevent and treat post-operatic abdominal distension. These are the uses. Improve the muscle strength and increase muscle responses. Route to per oral or intravenous and intramuscular. Onset of action 45 to 75 minutes oral. Then IV 4 to 80 minutes direct to enter. IM route 20 to 30 minutes. Duration oral is may take time 3 to 6 hours. IV short 2 to 4 hours only. IM and IV. Adverse dizziness, lacrimation, myosis, abdominal cramp, muscle spasm. Drug interaction, aminoglycoside. Don't admit aminoglycoside. Lincomycin, risk of neuromuscular blockade. Contraindication 2, mainly hypersensitive neostigmine, other anticholinesterase. Reverse non-depolarization, neuromuscular blocking agent. Myasthenia gravis is a neuromuscular disorder caused by abdominal immune system. These are some of the differentiation between physostigmine and neostigmine. Irreversible anticholinesterase bound to both only the ester side, which is all poison, is not at all used for any other therapeutic effect, only very rare. The coma patient only they tried for what is a mild dose of reversible cholinesterase enzyme inhibitor. Therapeutic uses very limited, only the toxicity, agriculture and household insecticide. The next one is the enzyme reactivators. Just now we have discussed, once the patient, once the victim, they attempt for suicide or unfortunately, maybe fortunately, they taken phosphorus poison, you have to absorb all the clinicism like amine, all the glandular secretion is increased, all the smooth muscle contraction is increased. I mean, cardiac heart in heart, decrease the heart rate, decrease the force of contraction, cardiac output is decreased, intercostal muscle is paralysis, finally death. What are the drug of choice? I mean, antidote for phosphorus poison. Antidote for phosphorus poison. The topic is named as enzyme reactivators. Under that, the heading is oxime and derivative. These oxime and derivatives are irreversible inhibition of phosphorylation of the ester side of the enzyme. Is combined with the phosphoryl group of this phosphorylate ester form soluble compound. Result, finally, reactivation of the enzyme. Then only it may be safe for your what you call the patient. The reversing of the neuromuscular paralysis due to organophosphorus, where the atropine is ineffective. The compounds are effective only the administration with a short time after poison. Late administration failed to produce expected result as may the time phosphoryl bound to enzyme get more stabilized. Oxime is mainly metabolized in liver and break down the product eliminated through renal. Oxime derivatives are adverse effective, local irritation, giddiness, blurring vision, neuromuscular blockade in high dose. Indication, treatment. Other drugs are also available. Predin 2 aldoxime chloride, 1 gram, route of administration IV. Severe cases 2 gram. If necessary, 2 gram, better you to choice is there. Second choice, diacetyl monoxime, diacetyl monoxime, dioxime, trioxime. That is the 3 different type of drugs are there. Dose 1 to 2 grams. Third one, obidoxime and derivative 3 to 6 milligram. Adrenergic neurotransmission, this topic already I discussed. I mean, neurotransmitter is noradrenaline and adrenaline. Postganglionic sympathetic nerve, noradrenaline release is there. This noradrenaline bound to major alpha receptor, very weak and minor beta receptor action. Another one, that is actually adrenergic adrenal medulla release of adrenaline. This adrenaline, which is having what we call alpha and as well as the beta receptor action, major beta and minor what we call alpha receptor action. These are all coming under catecholamine derivative. There's a pathway of adrenaline and dopamine, dopamine, noradrenaline, and adrenaline. So biochemistry already we are. You may know about this pathway. I mean catecholamine pathway. This catecholamine pathway, tyrosine, dihydroxyphenylalanine, phenylalanine to tyrosine, dihydroxyphenylalanine, dopa to dopamine, dopamine to norepinephrine or noradrenaline, noradrenaline to adrenaline or epinephrine. These are all catecholamine pathway. This is already discussed about the topic. Now, there is a flow chart, the diagram, what is the first one? How the noradrenaline is synthesized. It's hydroxylation of tyrosine, the rate limiting step, the tyrosine convert to DOPA, dihydroxyphenylalanine, DOPA to dopamine, that's a dopamine. This dopamine only enter into the vesicle and convert into norepinephrine. These norepinephrine protect from the degradation of vesicle. Transportation the vesicle inhibited by recepin. I mean catecholamine depletors. That is storage dip only, the recepin action is there. Third, number 3, release of neurotransmitter. Influx of calcium causes fusion the vesicle with the cell membrane. That process known as exocytosis. Release it on one side and blocked by guanethidine, and that these are all adrenergic neuron blocker. And the neuron itself blocked by guanethidine and beryllium. That is 3. Binding to receptor. There's an noradrenaline bind to alpha receptor, adrenaline bound to alpha and beta receptor. There, postsynaptic receptor is activated. I'm talking about 4. Synaptic says binding to neuronal transmitter. Then which is given a suitable physiological response. After that is metabolized. Metabolized by that is 2 enzymes. That 2 enzyme is named as COMT and MAO. COMT means catechol orthomethyl transferase and monoamino oxidase. Catechol orthomethyl transferase and monoamino oxidase. There is the 2 enzymes there. In the part, I will mention one more point. Antidepressant drug. And the antidepressant drug, one more classification is there. Monoamino oxidase, MAO. Monoamino oxidase enzyme inhibitor. What's the role behind? That is a depression patient. I'm talking about depression patient. Depression patient, there is exosynthesis and release of monoamino oxidase enzyme. Though dopamine, noradrenaline, and adrenaline are the 3 biogenic amines are immediately metabolized. It not reaches to your brain and other places. So the patient may be induced depression psychosis. There is a very good drug of choice is monoamino oxidase enzyme inhibitor. This is not indirectly related to one to another subject. So all the catecholamines are metabolized by 2 enzymes. One is catecholamine orthomethyl transferase, catecholamine methyl transferase. Another one more enzyme, monoamino oxidase. These 2 enzymes are metabolized. Again, there is a reuptake mechanism. Another one is some of the drugs are removed from the norepinephrine, release of noradrenaline, rapidly taken in the neuron. Reuptake is inhibited by some of the components, cocaine, local anesthetic drug. Another one is imiprimine, antidepressant. These are the mechanism. Key point, synthesis of noradrenaline take place the postganglionic nerve ending. These are all already we discussed. Right. Adrenergic receptor, alpha receptor, and the beta receptor. Alpha again divided into alpha 1, alpha 2. Alpha 1 is again divided into alpha 1A, alpha 1B, alpha 1D. Alpha 2 is divided into alpha 2A, alpha 2B, alpha 2C. Beta adrenergic is a beta 1, beta 2, beta 3. Beta 1 predominantly present in myocardium. Beta 2 is present in bronchial smooth muscle. Beta 3 is present in adipose tissues. Mechanism of the alpha receptor, execute response except small intestine. Stimulation of postsynaptic alpha 1 receptor, activation of phospholipase enzyme and cell membrane via G protein coupled receptor. Phospholipase, converted to diacylglycerol plus ionosyl triphosphate. These are all secondary messengers. Diacylglycerol and ionosyl triphosphate secondary messengers. Mechanism action already were discussed. They activate G protein gated potassium channel resulting membrane hyperpolarization. Alpha 2 inhibit, first one is activated, second one is inhibit. Inhibit voltage gated calcium channel which is mediated by GO. Then CNS, alpha 2A receptors are predominantly present in, and as well as inhibit noradrenaline release. Autoreceptor mechanism. And then sympathetic nerve ending suppresses sympathetic outflow brain leading to hypotension. Alpha 2A receptor CNS produces anti-nociceptive analgesic action effect, sedation, hypothermia, and hypotension. Mechanism action of beta receptor action is subdivided into beta 1, beta 2, beta 3. That is a GS. That is what we call G protein coupled receptor and as well as GS and activated by adenylyl cyclase enzyme. Increase intracellular cyclic AMP level and stimulate the enzyme protein kinase. Protein kinase phosphorylate in certain enzyme protein, then are activated by whole is activated followed by inactivated. There is autoreceptor mechanism. I mean modulator effect. The beta receptor simultaneously stimulated some inhibitor mechanism also. Inhibitor mechanism only branchial smooth muscle inhibitor mechanism. Heart excitatory mechanism, opposite effect. Alpha 1, some of the flow chart was location, blood vessel, pilomater muscle, radial muscle, heart and uterus. Constriction, contraction, heart. Alpha 2, pancreatic cell, platelet, nervous terminus. Decrease insulin release, aggregating the platelet aggregating factor. These are all agonistic, is clonidine, inhibited by yohimbine. Alpha 1 is stimulated by phenylephrine, methoxamine, inhibited prazosin. Alpha 1A, where it is located, heart, liver, cerebellum, cerebral cortex. And then predominant receptor causing contraction of smooth muscle. Alpha 1A, kidney, and the cerebral cortex. Then that is the most abundant subtype. Alpha 1D, platelet and cerebral cortex. These are the things over there. Beta 1 receptor, where it is present. Location is juxtaglomerular apparatus and heart. Juxtaglomerular apparatus, what is role is a renin. Renin converted to angiotensin 1, angiotensin 1 converted to angiotensin 2. Angiotensin converted into 3, 2 converted into 3. Next one is aldosterone. These are all renin pathway. Renin pathway. In this pathway, there is one more enzyme is that's named as angiotensin converting enzyme, ACE. It's an anti-hypertensive topic. The particular topic is that is angiotensin converting enzyme inhibitor, ACE inhibitor, ACE inhibitor. Next one, aldosterone antagonist, telmisartan, aldosterone. That is these are some of the next are there. Here, beta 1 agonist are dobutamine, blockers are atenolol and metoprolol. Beta 1 selective. Beta 1, beta 2 blocker is propranolol. Propranolol is what we call highly sensitive for asthma patient. Beta 2 the propranolol. Beta 2 is present in smooth muscle, bronchial smooth muscle, skeletal muscle, liver. Relaxation of bronchial smooth muscle, glycogenolysis and gluconeogenesis. Salbutamol, terbutaline. Blocker, propranolol. Beta 3, adipose tissue, lipolysis. These are some of the alpha 1, alpha 2. I don't have time. All those things I'm unable to cover. Sorry. Catecholamine, noradrenaline, dopamine, adrenaline, isoprenolol, or catechol nucleus. A double side, I mean OH. So this is the catechol nucleus. This catechol is a noradrenaline and dopamine sympathetic neurotransmitter. Adrenaline is a hormonal synthesized from adrenal medulla. Isoprenolol is a pure synthetic hormone. Beta receptor action. Non-catecholamines are ephedrine, amphetamine. Therapeutic classification: adrenergic used for rising BP, noradrenaline, metronidazole, phenylephrine. Adrenergic with inotropic effects: dopamine, dobutamine, isoprenolol. Central stimulant: amphetamine, dextroamphetamine, methylephedrine. Smooth muscle relax and non-selective: adrenaline, isoprenolol, and isosuprine. Selective beta 2 stimulants: salbutamol, salmeterol. These are all osmotic drug. Salbutamol, salmeterol, terbutaline. Anti-allergic drug, universal anti-allergic. Adrenaline is not an anti-allergic drug, but the allergic symptom is maybe act like a anti-allergic action. Adrenaline and ephedrine. Vasoconstrictor. Adrenaline, phenylephrine, xylomethrin. Appetite, fenfluramine, pentolamine. So the classification of adrenergic drug. Next one, this is catecholamine pathway. Already we have discussed. Heart also already discussed. These are all Dale's vasomotor reversal. Adrenaline is having alpha and beta receptor action. Beta 1 receptor increase heart rate, force of contraction and conduction velocity, increase myocardial oxygen consumption, cardiac output, and stimulate vasoconstrictor, enhance the conduction of across the AV node, may produce ventricular arrhythmia. Noradrenaline does not increase the heart rate, intact animal, but produce bradycardia due to compensated reflex vagal action. Myocardial effects are adrenaline, noradrenaline can be blocked by beta blocker like propranolol. Blood vessels, adrenaline, noradrenaline are constrict the blood vessels, skin and alpha receptor action. Adrenaline dilate the blood vessel, beta 2 receptor action. Adrenaline raises systolic pressure, cardiac action, lower systolic, diastolic blood pressure is a peripheral action. Both adrenaline and noradrenaline constrict the hepatic mesenteric blood vessel. Blood pressure, both response of noradrenaline on the rise in both systolic and diastolic pressure. Pulse pressure unaltered the mean of blood pressure increase, nor an induced hypertension associated with reflex bradycardia. Next one is so noradrenaline is having a biphasic response, alpha and beta receptor action. Alpha side skin and mucous membrane blood vessels are constricted, so rise in blood pressure. Beta receptor action, skeletal muscle blood vessels are dilated, so fall in BP. The Dale is one of the scientists. He only identified the Dale's mechanism. He only is identified with the help of dog blood pressure. With the help of ergot. Ergot is alpha blocker. Ergot extract, in the presence of ergot extract is done with one more experiment, dog blood pressure. Initially, a rise in BP followed by slight fall. That is maybe alpha receptor action, increase BP. Slight fall is what you call the decrease beta receptor action. Then you're going to block it by in the help of what you call ergot alkaloid, alpha blocker. That is blocked after administration of adrenaline, only fall in BP. Rise in BP is blocked by alpha blocker action, nothing but ergotamine block the alpha receptor in the vasomotor center. Complete blockade of vasomotor is stimulate and release of activation of the more beta receptor action, then only fall in is nothing but Dale's vasomotor reversal. Bronchial smooth muscle relaxation. Uterine, pregnant and non-pregnant may be aeration is a non-pregnant contraction. Pregnant, then maybe relaxation. GIT smooth muscle relaxation. Spinal muscle constricted. Other smooth muscle, pilomotor muscle, hair follicle contracted. Trigoral spinal muscle relaxation. Adrenal normal contracts splenic capsule. Then release of RBC, eye dilatation of the pupil. Metabolic action, adrenaline increase blood glucose level, enhance hepatic glucogenolysis, increase blood lactate level, break down the glycogen. Anti-allergic action, adrenaline prevent the histamine, is a mast stabilizing action. So the allergic time, adrenaline act like an anti-allergic action. Anaphylactic shock, very good use of adrenaline. Catecholamine is not administered orally. On inhalation, absorb systemic circulation. Metabolized by, already I discussed about the topic, catecholamine orthomethyl transferase and monoamine oxidase. Adverse effect, palpitation, thrombing, headache, tremors, ventricular apnea and anginal pain may be. Ischemic cardiac disease. Preparation, 1:1000 solution. Adrenaline. They're in the form of units. Dose and administered 0.2 to 0.5 ml. Subcutaneous or IM. Dead slow, what you're going to administer, students, listen, the adrenaline directly not administer. Very, very slowly you to administer adrenaline. Take care. Dead slow. I mentioned the word dead slow. Slowly administer, or otherwise, sudden collapse, sudden death may be occur. Immediately death may be. நீங்க சட்டுன்னு போட்டு திரும்பிட்டீங்கன்னா பாடி உழுந்து கிடக்கும், செத்து போயிடுவாங்க, காப்பாற்ற முடியாது. அதனால அட்ரினலின் போடுறப்ப மட்டும் ரொம்ப ஜாக்கிரதையா போடுங்க. போடணும்னு அட்வைஸ் பண்ணுங்க. ரொம்ப ஸ்லோவா போடணும், ரொம்ப டெட் ஸ்லோவா போடணும். அதுல இது பண்ணக்கூடாது, ரொம்ப இதெல்லாம் ரிஸ்க் எல்லாம் எடுக்கக்கூடாது. இட்ஸ் வெரி டேஞ்சரஸ் ஒன், பட் இட்ஸ் லைஃப் சேவிங் ட்ரக். உயிரை காப்பாத்திடும். ஃபாஸ்டா போட்டு டாக்ஸிக் ரியாக்ஷன் இஸ் ஓகே. Adrenaline inhalation, noradrenaline injection, 0.2 solution, 2% solution, route of administration, IV. Only a noradrenaline only you administer IV infusion. Therapeutic, anaphylactic shock, bronchial asthma, cardiac pulmonary rescue, then control the hemorrhage along with local anesthetic. Therapeutic and noradrenaline, shock, elevation of BP. Isoprenoline is a synthetic drug, non-selective beta agonist property. Is a catecholamine derivative. Stimulate heart and causes tachycardia. Lower the peripheral vascular resistance. Is relax the bronchial smooth muscle, mainly relaxation. Asthmatic patient, very good drug of choice is there. The metabolic action similar to adrenaline. Catecholamine and monoamine is metabolized. Preparation dose, isoprenoline tablet, 10 milligram available. Dose 10 to 10 milligram sublingual route of administration. As well as also available in the parenteral route, IV infusion 2 milligram per ml. Therapeutic use of Stokes-Adams syndrome, sudden transient episode of syncope, occasional future seizure, shock due to beta blocker overdose. Dopamine is a central neurotransmitter. That neurotransmitter is a catecholamine, third precursor of noradrenaline. Central neurotransmitter Excess synthesized and released more, it may be hypertension. Also may be aggravated. Then this is induced schizophrenia. And excessive blockade of dopamine and dopaminergic is what do you call the receptors is a Parkinsonism. Parkinsonism. Dopaminergic receptor, presynaptic brain, postsynaptic present in peripheral. D1 like action include D1 to D5 mechanism action. D1 receptor increase cyclic AM synthesis. D2 receptor decrease in cyclic AM synthesis. These are all some of the pharmacology action. D1 receptor is in hypothalamus, exert autonomic control. D1, D2s are limbic system, mood and emotion. D2 receptors are corpus striatum, exert motor control. D2 receptor, pituitary inhibition, the prolactin secretion. Peripheral D2 receptor mainly in heart muscle. Dopamine 5 to 10 microgram per kg. Dopamine, other inotropic drugs. Okay. The functions and the distribution function. Adrenergic these are all ephedrine and drug ephedrine, non-ephedrine. Resemblance of the ephedrine is the noradrenergic action, alpha major action, alpha receptor major action obtained from ephedra synca. The sympathomimetic drug, alpha and beta receptor action is there, but more alpha receptor action. Ephedrine rise in blood pressure, but repeated administration induced tachyphylaxis. Ephedrine relaxes bronchial smooth muscle, stimulates CNS. Adverse effect like immune catecholamine, bronchial asthma, nasal decongestion, hypotension, Stokes-Adams syndrome, mydriatic effect, narcolepsy. Amphetamine is a CNS stimulant drug. Weak beta receptor stimulation. Mechanism action, amphetamine in all CNS effect indirectly release the biogenic amine stored in nerve ending. Therapeutic classification, vasopressor agent, noradrenaline, dopamine, ephedrine, phenylephrine, methoxamine, cardiac stimulant, adrenaline, isoprenaline, dobutamine, bronchodilator, adrenaline, isoprenaline, salbutamol, terbutaline, salmeterol, and formoterol. Nasal decongestion, phenylephrine, xylomethane, pseudoephedrine, and nafacillin. CNS stimulant, amphetamine, dexamethasone, methadone, anoractin, fenfluramine, and dexaferine. Uterine relaxant, retrodrine, isoprene, salbutamol, and terbutaline. These are all blocker. Another name is antihypertensive agent. Alpha receptor blocker, beta receptor blocker. There I am going to divide it into alpha adrenergic blocker, further into alpha 1 blocker, alpha 2 blocker, again alpha as well as beta receptor blocker. There is a non-selective alpha blockers are they classified beta haloalkyl, dibenamine, phenoxybenzamine. Dihydrogen ergotamine, ergosine, ergotaxamine. Imidazoline derivative, tolazoline, pentolamine. Selective alpha 1 blocker, phenazoline, prosocine, tyrosine, toxaverine, endoramine. Selective alpha 2 blocker, yohimbine. Non-selective dibenamine and phenoxybenzamine is a reversible blockade of the effect. These are all produces meiosis effect. Natural dihydroxyl, ergotamine, ergosine, ergotamine, and dihydrogen. Dihydrogenated alkaloid reduced blood pressure, alpha blockade, vasoreceptor, and adverse effect. Next one, quinazoline and derivative. Quinazoline and derivative. These are all also maybe prosocine. Is mainly alpha 1 receptor blocker. Giddiness, adverse effect, drowsiness, nausea, diarrhea, palpitation. Yohimbine is a alpha 2 receptor blocker. Beta receptor blocker, both beta 1, beta 2, propranolol. Cardio selective blocker, asbutol, atenolol, metoprolol, esmolol. Beta selective 1 blocker, because asthma patient don't administer beta 1, beta 2 receptor blocker. Selective beta 1 blocker only. Non-selective beta blocker, propranolol, pendolol, timolol. Beta blocker with additional alpha blocking agent, carvedal, salividol, labetalol. Alpha beta blocker, labetalol, carvedal. Adrenergic antagonist, these are all adrenergic neuron blocker. I mean the neuron directly bound to the neuron. Once you go into administration, stand minimum 1 week. I mean guanethidine, bethanethidine, bathanacar. Another one is catecholamine depleters. Example, serpine. Catecholamine depleters. Catecholamine means storage depot, adrenaline, noradrenaline, dopamine are stored. Once you go to administer adrenergic blocker, the pressure may be fall down, but further the catecholamine going to slowly release adrenaline, noradrenaline, dopamine, again increase. The confusion is there. Reserpine is a very good effect for catecholamine depleter. There is a storage depot completely blocked. Further release of catecholamine is blocked by reserpine. These are the schematic representation. Reserpine is a plant alkali, Ravalfia serpentina, reserpine used for 2-in-1 action. One is isoprenia, antipsychotic drug, another one is antihypertensive agent, classifying under catecholamine depleters. Same antipsychotic drug, major schizophrenia. Schizophrenia is a very good drug of choice. Is there. Guanethidine is adrenergic neuron blocker. Once you going to administer, minimum of the peak hours 8 hours and withstand in our body 7 days. 7 days is a stand. Beryllium, the bromobenzyl quaternarium ammonium compound. Why I'm going fast mean I have 50 MC question is there. If time permits, I'm going to discuss it. Okay. Just a minute. Madam, excuse me. Madam, இது மட்டும் சிக்கு ஒண்ணே ஒண்ணு வச்சிருக்கேன் அது மட்டும் கொடுத்துறவா
கொடுத்துருங்க சார் கொடுத்துருங்க
ஓகே சரி மேடம் எம்சிக்குல அது கொஞ்சம் ஒரு ஃபாஸ்டா பண்ணணும் அதே நான் முன்னாடி சொன்னா இல்லீங்களா ரொம்ப வாஸ்ட் சிலபஸ்
ஆமா சார் பரவாயில்ல சார் எம்சிக்கு கொடுத்துருங்க அசைன்மெண்ட் மாதிரி கூட செய்ய சொல்லுங்க இல்ல எக்ஸ்பிளைன் பண்ணாலும் பரவாயில்ல இருக்காங்க ஸ்டூடண்ட்ஸ் ஒரு 50 ஸ்டூடண்ட்ஸ் இருக்காங்க
ஸ்டூடண்ட்ஸ் this is your highlight for GPAT examination. I think for me a maximum I covered. How many students are you then and understanding about this topic? I don't know. But anyhow, all the best. These are the things are test yourself. I just will discuss one by one. Cholinomimetic drug are not used in choice. Glaucoma, myasthenia gravis, post-operative atony, partial heart block.
Kalaiselvan sir, இன்னும் ஷேர் ஆகலன்னு நினைக்கிறேன் ஸ்கிரீன். இப்பதான் ஆயிட்டு இருக்குன்னு நினைக்கிறேன். கண்ணன் சார்.
ஷேர் ஆயிடுச்சா சார்?
இல்ல மேடம் சார் அதே மாதிரி ஒரு வாட்டி க்ளோஸ் பண்ணிட்டு திருப்பியும் ஓபன் பண்ணி பாருங்க சார்.
ஓகே சார் நான் வரேன் சார்.
ஸ்டாப் ஷேர் கொடுத்து
வரேன். டிஸ்ப்ளே அத பாருங்க
இன்னும் வரல Okay, sir.
Okay, sir.
Share ஆகுது சார் கலெக்ஷன் ஷேர் ஆகுது.
Okay. Students, மேடம் ஸ்டூடண்ட்ஸ் இப்ப क्वेश्चन கேட்டா சொல்லுவாங்களா அவங்க ஆடியோ வருமா ஓபன் பண்ண முடியுமா பண்ண முடியாது
இல்ல சார் அவங்க ரிப்ளை பண்ண முடியாது சார்.
ஓகே சார் அவங்க அந்த क्वेश्चन and answer வேணா ஆன்சர் பண்ணிட்டு போட்டோம். ஓகே நான் அதோட ஃபாஸ்டா முடிச்சறேன் 15 நிமிஷம்
சாட்ல வேணா போடுவாங்க பட் எவ்வளவு அளவுக்கு தெரியும் தெரியல உங்க சாட் பாக்ஸ்ல வேணா போட சொல்லுங்க அது நேரா ஆகும் ஒவ்வொருத்தரா யோசிச்சு.
ஓகே மேடம் நாம ட்ரை டு கம்ப்ளீட் இட் மேடம்.
ஆமா சார் நீ போடுங்க சார். ஓகே
ரைட் there is MC question cholinomimetic are not used in 4 choices are glaucoma, myasthenia gravis, post operative atony, then partial heart block. The correct answer is partial heart block. Cholinomimetic drug is not used for partial heart block. Next one, LID retention is caused by IMO protest, lanthoprotect, primodine, and aproclodine. The drug of choice is actual drug is aproclodine. Next, third question, which of the following drugs should not be given for patient with acute angle closure glaucoma? Glaucoma is a drug of choice, cholinesterase enzyme inhibitor, but it should not be given the patient with the acute angle glaucoma. You are not given. The drug of choice, example, pilocarpine, clofazine, and prolactin. Exact answer is clozapine. I mean B. Next one, which of the following anti-glaucoma mediation of unsafe in infants? That is actually infants is unsafe. Example, what you call choice A, temolol, brimonidine, lentosport, and dorsoport. Exact answer is B. B. Brimonidine. Brimonidine. Next one. The following drugs are following effects are seen with the cholinergic muscarinic receptor stimulation. All the following effects are seen in the cholinergic receptor, except. So already we discussed about the topic. Sweating is there, a rise in blood pressure is there, bradycardia or urination. Which one is correct? Except rise in BP. Rise in is not rise in BP. Okay, that's one because fall in BP. Actually, opposite effect. Next one. Agonist action, which of the following adenoreceptor results in the reduction of secretion? Agonist action of following adrenergic receptor result in reduction. So that is we are given beta 1, beta 2, M2, alpha 2. Agonist action, adrenergic receptor. Which one is best? Correct answer. Alpha 2 receptor. Alpha 2 receptor, I mean B. Next one. Drug of choice in the treatment of acute organophosphorus poison. Acute organophosphorus poison. Atropine, parallaxin, neostigmine, and dobutamine. Atropine is a universal one, but one side is maybe we are talking about obidoxim, paradoxim, and other things are there. But acute level, there is the first choice is atropine followed by paradoxim. There is some confusion is there, right? Next one, a patient compliance, 8th question. Muscle weakness, 1 point. Reverse for administration of neostigmine, 2nd point. Because what's the reason began? It block action of acetylcholine, interfere action of monoamine oxidase, or interfere with the carbonic anhydrase enzyme, or acetylcholine esterase. Which one is correct choice? That is interfere with the action of acetylcholine esterase. So interfere with the acetylcholine esterase. Next one, organophosphorus compound. Organophosphorus compound. Organophosphates bind to, already we discussed about the topic, that is two side already discussed, anion side, cation side. Reversals are anion and cation side. Organophosphorus compounds are only ester side. So which one is correct? Which one is correct? So ester side, the B is correct. Next one is 2-PAM is a paradoxin is useful in the treatment of paracetamol overdose, DTP poison, DTT poison, and malathion poison. This is malathion, parathion, TIC20, these are all poison. I mean phosphorus poison. Mainly this is used for phosphorus poison. This is coming under irreversible choline esterase enzyme inhibitor. Irreversible choline esterase enzyme inhibitor. Next one, drug of choice in the treatment of myasthenia gravis. D-dibochlorine, hexamethonium, neostigmine, and galamine. Which one is correct? That is, neostigmine is the exact answer. Next one, alpha 2. Spelling mistake is there, alpha 2. Alpha 2 agonist is used for glaucoma. Tamil Nadu GPAT examination 2018, they were asked. Guanakar, Gatanakar, Guapensin, Brimodine, and Timosine. It's a Brimodine, Modine is a drug of choice in the treatment of alpha 2 agonist for exclusively. Oral poison, carbamate, insecticide is an insecticide. Dash may be the hazardness, parallaxin, atropine, magnesium sulfate as a purgative, and gastric levase flavonode. What it is the drug of choice mean? Paradoxim. This is a very good antidote for oral poison. 14th question. A patient presented emergency with admitted tachycardia is having, hypothermia is having, bronchial dilatation, constipation. The person likely to suffering from overdose of atropine, organophosphorus compound, mushroom, or paracetamol. Which one is correct? Atropine. Atropine high dose may induce tachycardia, hyperthermia, bronchial dilatation, constipation. Next one, which is 5th question. Which is the shortest acting mydriatic? The GPAT exam 2010 they are asking question. Mydriatic effect. Shortest mydriatic action. No, I'm not asking about longest. Shortest. atropine, troponamide, and cyclopentone, then homotropine. Troponamide B, troponamide. Another one. Fundoscopy, short-acting mydriatic used for fundoscopy examination. Which one is actually anticholinergic drug is there? Which one is actually drug of choice is there? Mean troponamide, TROPA, tropocamide, tropocamide. Next question. Pyranazepine is at which receptor action? Muscarinic, nicotine, alpha, or beta? Pyranazepine. The muscarinic receptor action, Which of the following, 18th question, concentration epinephrine, adrenaline, does not correspond to respective route of administration. We are not administered the particular route in the concentration. 1:10,000, 1:1,000, 1:1,000 for inhalation, intramuscular, and 1:1,000 for subcutaneous. Inhalation route because more toxic. ரொம்ப டாக்ஸிக்கா இருக்கிறதுனால டோஸ் குறைச்சி கொடுக்கணும். அடுத்தது which of the following drugs long acting alpha beta 2 agonist? Salmeterol, long acting, mainly for asthmatic patient. Asthmatic patient கொடுக்கக்கூடிய drug. Beta 2 agonist drug. I'm not saying antagonist, agonist. Next one, drug used to perform stress echo. Echo எடுக்கிறப்ப ஒரு stress தேவைப்படும். Which one is the induced echo? Perform this. Thallium, dobutamine, adrenaline, and dobutamine. In these cases, but you administer dobutamine. 25th question. Dopamine preferred in the treatment of shock because also maybe UP examination gate. Another one is a DPG. 2010 also both places they are asked. Dopamine preferred in the treatment of shock because of renal vasodilatory effect, increased cardiac output, peripheral vasoconstriction, prolonged action. Renal vasodilatory effect. Then only maybe the shock may be come back to the renal clearance is there. Then the shock is slowly going to release. Depression occur. The side effect of due to reserpine, propranolol, morphine, and amphetamine. Just now we discussed few minutes back. Depression topic already we discussed about the topic. Reserpine, propranolol, morphine, amphetamine. Which toxicity? Depression side effect is reserpine. It's a catecholamine depletors. Excessive dose that may induce depression. Depression is named as reserpine reversal. Simultaneous administration of reserpine கூட சேர்ந்து எடுக்கிறப்ப antihypertensive எடுக்கிறப்ப இந்த மாதிரி ரிவர்ஸ் ஆகும். ரிவர்ஸ் ஆகுதான் பேர் சொல்றாங்க. reserpine reversal side effect. Next one is contraindication of beta blocker. Contraindication, this is also already pointed out. Contraindication for beta blocker: asthma, heart block, hypertension, arrhythmia. Mainly for there is both correct. Asthma also, heart block also. Contraindication for heart block, then asthma. That's two choices there. Just which are the following third generation of the beta blocker? Third generation. Propranolol, timolol, nadolol, and navitol. Alpha blocker. Navibiol. Okay. 21 question. Vasopressor in pregnancy. If pregnancy time, vasopressor is the drug of choice in the pregnancy time. Ephedrine, phenylephrine, methoxamine, methamphetamine. Never the ephedrine. Ephedrine is a drug of choice. Next one, a patient. Harish is the name of the patient. Emergency after receiving penicillin. His diagnosis is anaphylactic shock. He received immediately shock is there. Which of the following only life-saving measurement for treatment? Once the penicillin injection, immediately they produce anaphylactic shock. Within few minutes, the patient may be, the patient Harish may be collapsed. Which drug of choice is there? Intravenous administration hydrocortisone, intravenous administration adrenaline, intramuscular adrenaline, intravenous glucose saline. Which one is correct? That is adrenaline. Intramuscular adrenaline or subcutaneous. Subcutaneous you have to administer. Next one, a drug is shown activate dopaminergic D1, D2, adrenergic alpha 1 and beta 1, but not beta 2 receptor. Which of the following can be drug being talked about? Activated dopaminergic D1, D2, either dopamine or dobutamine or methoxamine or phenylephrine. That is dopamine A. Beta-2 selective agonists are often effective in treatment of angina, asthma, delayed labor pain, or all of the above. All of the above. That is B, asthma. B, asthma. All right. Which of the following selective beta-2 antagonist that's already displayed? Selective beta-2, beta-2 antagonist, esmolol, betoxolol, betoxamine, and celiprolol. This is betoxamine. Heart rate is decreased by propranolol, isoprenaline, propranolol. All are alpha blocker except atenolol, prazosin, intromine, and altenolol. Atenolol is a beta blocker. Then next one is all of the following statements of dopamine are true except increase GAT, then positive inotropic effect, and improve the renal perfusion causes vasoconstriction. A causes increase ischemia. Most potent cardiac stimulants are adrenaline, noradrenaline, ephedrine, and other salbutamol. Which one is more potent than adrenaline? Is a potent drug. Next one. Is it share? Okay. Which of the following drugs are increased GAT motility? Neosigmin is a increased GAT motility. Glucopiridine, atropine, neosigmin, fentanyl. Drug of choice in the treatment of anaphylactic shock. Just now we discussed in the point. I mean, adrenaline. Adrenaline is a drug of choice. Paroxysm. This is actually enzyme reactivator. Enzyme reactivator. It's inactive in the which type of poison? Edala the poison only there. Carbonyl poison. Carbonyl poison is inactive. Which of the following does the most effective in control orthostatic hypotension? Clonidine, fluocortisone is small, and phenylephrine. Fluocortisone is drug of choice. Next one. Alpha blocker. Alpha blocker useful for the BPH. Which one is correct? Pentolamine, prazosin, tolazarine, which is 4 among the 4, which one is the drug of choice? Prazosin is the drug of choice. Beta blocker contraindicated to decompensated congestive heart failure, asthma, variant angina, all the above. All the above. All the 3 is beta blocker is contraindicated. Botulinum toxin. Already we discussed about the topic. Secretion of acetylcholine, synthesis of acetylcholine, inhibit the acetylcholine muscle nerve block. That is inhibit the acetylcholine release. Botulinum is the inhibit the further release of this one. Atropine is added the commercial preparation of diphenoxylate. Potentiate antispasmodic effect, reduced excretion. In a drug of choice, first C, to prevent overdose of the discourage of the opioid analgesic. Opioid analgesic drug. Next one, beta adrenergic drug. Beta adrenergic blocking should be avoided patient with renal failure. Renal failure is not administered, not advised. Atenolol is an exact answer. Next one, no, 10 more is there. Drug of choice for bradycardia. Which one is due to beta blocker overdose? Which one is better choice? That is atropine. Which of the following? Which of the following? Following is a selective alpha 2 antagonist. This is also already discussed. Yogamine. Next one is atropine most sensitive to mucous, pharyngeal, heart, pupil, GAT motility. That is mucous pharyngeal secretion. Atropine is most sensitive to mucous pharyngeal secretion. So slurred speeches are difficult to swallowing. Oral cavity voice. Atropine is used for all except glaucoma, Midriatic, cyclopedia, pre-anesthetic medication. Glaucoma the contraindication. Next one, Midriatic drug has short duration of action. Cyclopentanol, tropinate, and homotropin, atropine. Best one is tropicamide. This is also already discussed. All the following are used for atropine poison except paradoxine, atropine, activated charcoal, naltrexone. Fourth one. Which of the following is used for the anaphylactic shock? This is adrenaline. Adrenaline. The universal antidote. The anaphylactic shock. Universal one. Sweating is the result excretion mediated through adrenal hormone, sympathetic adrenergic, parasympathetic, and sympathetic. This is actually sympathetic cholinergic nerve ending. Sympathetic cholinergic nerve ending. Okay. Thank you, one and all. Thank you, one and all. Thanks for dear participants, members, and the GPAT team and others. Very much patiently you listen, everybody. Thank you very much, madam. Thank you very much, madam.
Thank you, sir. Thank you, sir.
Thank you, sir.
Thank you for your extensive talk on ANS and the questions, nearly about 50 questions.
Yes, madam. Yes,
Really, the students are benefited.
Yes, madam.
I would like to thanks to the students also, those who are presented till the end.
Yes, thank you very much. Thank you.
Thank you, Madam Meenakshi, Madam Kannan sir, Vinayamurthy sir, and Uma Mageshwari, Madam. Thank for the entire team.
Thank you, ma'am. Thank you, sir.
Thank you, sir. Thank you, madam. Thank you.
Thank you, madam. Thank you, madam.
Lecture, sir. Thank you.
Thank you.
ரொம்ப எஃபோர்ட் போட்டு நல்லா பண்ணிருக்கீங்க சார். ஸ்லைட் நல்லது. சுமதி மேடம் ஆல்சோ ஸ்லைட்ஸ் ஆர் வெரி குட். போத் ஸ்பீக்கர்ஸ் டுடே they put a more effort and dedication in the preparation, sir. The slide itself shows it.
Thank you, madam. Thank you, madam. Thank you, madam. Thank you.
I would like to thank
vast syllabus, you know.
Sir, one more. Can I ask a question, sir?
என்ன மேடம் சொல்லுங்க மேடம்.
இல்ல ஒரே ஒரு கொஸ்டின். அந்த பாட்டிலினம் டாக்ஸின் சொன்னீங்களா, அது கூட ACH வருமா?
ஆமா மேடம். ஆமா. அதாவது டாக்ஸிக் டோஸ் கொடுக்கறப்பதான் அது மாதிரி வரும். அது that it itself may be a release of a cell killing. அதுவே அது தன்னைத்தானே ரிலீஸ். This is itself is released.
Nowadays for cosmetic purpose யூஸ் பண்றாங்களே.
ஆமா மேடம். வீக்கர் எல்லாம் வீக் பார்த்தீங்கன்னா 0.1 0.2 because it may be induced sweating secret and sweat opener ஆகுது. sweat open
that
ஓபன் பண்ணி temporary sweat glands ஓபன் மேடம். ஓபன் பண்றப்ப
பேசணும்.
ஓகே
மேடம். சார் கலெக்ஷன் சார் அந்த ஸ்லைடு வந்து பசங்க எல்லாம் சாட் பாக்ஸ்ல போட்டுட்டாங்க ஷேர் பண்ண சொல்லி.
ஓகே மேடம்.
முடிஞ்சா பாருங்க சார் எப்படின்னு.
இல்ல நான் அப்படியே பசங்களை அனுப்பிடுங்க மேடம்.
சரி சார். IPGA
MCQ வேணுமா அந்த தியரி வேணுமா மேடம்?
ரெண்டுமே MCQ கேட்டிருந்தாங்க, அந்த இதே கேட்டிருந்தாங்க. உங்க பிரசன்டேஷன் தியரி வந்து போடும்போது சாட் பாக்ஸ்ல வந்து PDF ஃபைல் எங்களுக்கு போட்டீங்கன்னா நல்லா இருக்கும்னாங்க.
ஓகே மேடம். ஓகே நான் அனுப்பிடுறேன்.
நம்ம குரூப்லயே GPAT குரூப்லயே போட்டு விட்டுருங்க சார்.
ஆமா சார், குரூப்லயே போடலாம் சார். நீங்க
நான் குரூப்ல போடுறேன் மேடம். அது வந்து அந்த பசங்களுக்கு நீங்க இது பண்ணி
சரி சார், ஷேர் பண்ணிக்கிறோம் சார். Thank you சார். Thank you சார். Thanks to you sir, Kalaiselvan sir. Short timeல accept பண்ணி நல்லா பண்ணீங்க. Thank you and thanks to Sumathy madam also for their excellent presentation.
Thank you madam. நான் உட்கார்ந்திருக்க இடம் தெரியுதா மேடம்? மொட்டை மாடியில உட்கார்ந்திருக்கிறேன்.
நல்லா தெரியுது.
இது எங்க மொட்டை மாடியில தான் உட்கார்ந்திருக்கேன் மேடம். இது டிபார்ட்மெண்ட் தான் கிளியரா இருக்கும் உங்களுக்கு. மொட்டை மாடி அதனால எனக்கு டவர் கிடைக்கல மேடம்.
ஆமா கரெக்ட். அதனால தான் நல்லா இருந்தது சார். இது கரெக்ட்டா
எல்லா மரம் சுத்தி மரம் எல்லா மரமும்
சூப்பர் சார்.
Thank you madam. Thank you.
Thank you Mr. Kannan and Mr. Malar Kannan. Thank you sir.
Thank you Kannan sir. Thank you Meenakshi madam.
Thank you sir. Thank you sir.
Madam. Thank you. Everybody, IPGA and MMC team. Thank you very much.